Welcome to yet another research Q&A organized by Tinnitus Quest. This one's going to be a little bit different. Instead of having a live audience asking their questions live, we have Anthony here to interview Dr. Dirk de Ridder, which allows us to get a bit more deep and technical than in the first three Q&As. So Dirk is actually here for the second time to do this follow-up Q&A, and thank you, Dirk, for joining us again. The trigger for this was that Dirk's first Q&A raised a lot of questions, and some people are really deep into the research, wanted to know more. And in these kind of live public Q&As, it's difficult to do that. We can't get too deep and technical because we'll lose 90% of the audience. So this follow-up Q&A is, if you will, the nerd edition. And I mean that in a good way. It's for those of you out there who like to get deep into tinnitus science. And since I don't feel well positioned to do that, that's why we have Anthony here. And Anthony, I know you have prepared your questions very diligently, but not just by yourself. You got input from a lot of other patients who are deeply interested in tinnitus research. So in a few moments, I'll hand it over to Anthony to interview Dirk, and I will just be here as moderator. I'll keep the time. Maybe I'll ask some questions for clarification, but otherwise I will leave it to Anthony. Before we start the interview, however, I wanted to take the opportunity to clarify something. Our first three Q&As seem to have raised the impression among some people that the researchers we were featuring in the Q&As who are affiliated with Tinnitus Quest are also being funded by Tinnitus Quest, and that is not the case. Tinnitus Quest is still in the fundraising phase, so we're not actually funding any research yet. We hope to open up our grants program in early 2025, and we specifically hope to fund research into experimental new approaches to silence tinnitus conducted ideally by young researchers with fresh, out-of-the-box ideas. And the grants will be open for any qualified researcher to apply. The reason we have people like Dirk on board is with his, decades of expertise in the field, he will be helping us to assess the quality of those applications and make sure that money gets spent in the right way. So the purpose of our Q&As is really to give the public a chance to directly engage with researchers and ask their questions, because there's so little public communication about ongoing tinnitus research. And it's researchers like Dirk and Hamid and Bertold who are affiliated with Tinnitus Quest who have been very kind to spend their time engaging seriously with the public and answering their questions. Not because they claim they have all the answers. I mean, if we had all the answers, we wouldn't need an organization like Tinnitus Quest, but really, in the spirit of openly sharing their knowledge and their deep understanding of tinnitus science based on their decades of research experience and bridging that gap between patients and researchers. Okay, so without further ado, let's get into the Q&A. I will introduce Dirk, even though I've introduced him in prior Q&As. Maybe some people are listening for the first time. And then, Anthony, I will ask you to introduce yourself. So Dr. Dirk de Ridder is a neurosurgeon. He has been involved in tinnitus research for over 20 years, especially brain research with implications on neuromodulation. As a professor, he conducts his tinnitus research at the University of Otago in New Zealand, and as a doctor, he runs a neuromodulation clinic specialized in tinnitus in Ghent, Belgium. That's called Brain. But it somehow has the number 3 on it, so I don't know how to pronounce that. Anthony, would you like to introduce yourself to the viewers?
Absolutely. So my name is Anthony, as you all know, and I am the founder and owner of the Tinnitus Labs YouTube channel and Discord server, where we aim to connect the brightest minds of our community together to research and to discuss these conditions. I may also mention that I am a PhD student in neuroscience with a special focus on spike timing-dependent plasticity, which we will discuss later today. Well, I guess I'll go ahead and pass it to Dirk. Let's get started.
Well, good day, everybody. So many questions that we get asked is, Why is something not more studied? And this could be hyperacusis, misophonia, specific involvement of somatosensory tinnitus, etc. Well, the reason is relatively easy to explain. If you just look at the amount of publications, for example, in cancer and in tinnitus, then we see that up to now, there have been 17,000 PubMed listed publications on the topic of tinnitus since ever. For cancer, that's 5,200,000. And for pain, which is very similar to tinnitus from a neuroscience perspective, there is more than 1 million publications. So there is more than 65 times more pain publications than tinnitus publications and 300 times more cancer publications than tinnitus. Now, if you want to relate that to the amount of money funded for research, then what you can see is that tinnitus has probably, because these are more estimates than real numbers, for tinnitus has been around 200 million since the first study on tinnitus. Now, if you look at pain, then this amounts to about 20 billion. So there is 100 times less funding for tinnitus research than for pain research, even though the amount of people having chronic pain is about 20% of the population, and the amount of tinnitus patients amounts to about 15%. So even though it's close to being equally prevalent, the amount of funding for pain is dramatically more, 100 times more than the amount of funding there is for tinnitus. Which means, of course, there is less people attracted to the field of tinnitus research because it's so much more difficult to find money to conduct research. Which also means that if you only have a very limited amount of funding, that you start with what appears to be the most important or most relevant topic, and that X, which could be hyperacusis, which could be misophonia, which could be the influence of some specific ion channels or some neurotransmitters, is simply not being done because there's no research money to it. And just to put it into real perspective, if you look at cancer, there is a $50 billion research funding per year. Now, 50 billion per year is very different from what you have for tinnitus. And if you look at it, there is 800 times more money for cancer research than for tinnitus research. So it's clearly underfunded, and that's why there has been not that much research as we would love to being conducted in tinnitus.
All right. Can I ask some questions then?
Yes, certainly do.
Okay. Well, I think that something that I and our community is kind of curious about is, specifically, you explained the reasons quite well, but what we don't really understand is what specifically was the cause. Of the profound interest, for example, of pain, even though the percentages of the population are pretty much the same. Could it be that some, for example, psychological interventions have taken the forefront and been considered to be more of an adequate measure instead of actually studying the deep neurophysiological mechanisms?
Yes, I think the main reason is that also from a research point of view, we have what we could call a physiological pain pathway. So you can study pain, acute pain, very easily by inflicting pain on a guinea pig or on a human participant in a study and measure how pain is processed physiologically. Chronic pain, however, is pathological, and so people could study acute pain and then speculate about how this acute pain could move into chronic pain. Yet for tinnitus, there is no tinnitus pathway. There is no physiological tinnitus pathway. So this made it a lot more difficult. But both tinnitus and pain share the fact that they're basically entirely subjective entities, and that they're difficult to study. But at least in acute pain, everybody knows what acute pain is, but not everybody knows. Well, 85% of the people don't know what tinnitus really means. And therefore, unfortunately, tinnitus is a lot less sexy from a research point of view and from a conceptual point of view, because everybody knows what pain is, because everybody has had acute pain once or more in their life, whereas for tinnitus, that's not the case because we just don't have a tinnitus pathway.
Oh, okay. That's understandable. That's a great explanation. So, as I understand, what we would have to do is to establish an objective measure of tinnitus, which is something that I know Bionics Institute is working on with a non-invasive brain imaging technique. And I truly myself believe that establishing this objective measurement will, I think, boost the potential for tinnitus research very much. Okay, well, I guess let's go ahead and move on.
And I fully agree with you. And I think we're also looking for that, trying to use EEG to look at tinnitus and then correlating it to loudness estimates as well as suffering estimates. So the goal is to have a two-way objective measure, just like you have a blood pressure to look whether somebody has hypertension or not. We want to look for brain signature using simple EEGs because that's cheap, that's very easily doable, and have two different approaches. One approach is just to be able to say yes or no, there is tinnitus. Yes, there is tinnitus, or no, there is no tinnitus. The second question, however, is if there is tinnitus, can we also look for a brain signature of how loud it is perceived by the person and how much suffering does it cause? But in order to be able to do that, using artificial intelligence, you need very large databases that are collected with the good EEG machines, and you need all those clinical data, which up to very recently did not exist. And so together with Professor Jae-Jing Sung from Korea and our database, we will make an attempt to look at close to 2,000 EEGs and to find, hopefully, this diagnostic signature of tinnitus, as well as a sign of how loud and how bothersome it is. And I fully agree that once we have that, that would allow, first of all, it would convince people that, yes, tinnitus is between your ears and your brain, and that it is something that is objectively measurable and not only subjective. And this, of course, would then allow companies to use pharmacological approaches and say, look, this works because the tinnitus might still be there, but it drops in its intensity, loudness, it drops in its suffering based on these objective reasons. So I fully agree with you that it will boost not only clinical practice, but also research.
So, another question that has been raised numerous times in the tinnitus community is that some specialists believe that there is no correlation, or there's no direct correlation to the suffering caused by tinnitus and the tinnitus volume. I guess we could also include reactivity here. I wanted to hear Dirk's opinion on that.
Yeah. So this is a very interesting question and comes to the core of what is suffering and what is the loudness perception. So we know we've studied this in pain in more detail, and we would like to study it as well in tinnitus, but we just lack some data. So in pain, we know that if somebody has pain with this, that can lead to suffering. It doesn't have to lead to suffering, and it can lead to disability. And so we looked at a large group of patients who came to a neurosurgical unit because of pain, and we did some statistical testing using cluster analysis, which means you look at in the total group of people who present at a neurosurgical clinic for pain, do we have different subgroups that can be statistically separated from each other? And what we found is there's actually 3 groups. One just has pain, no suffering, no disability. And their score of pain was less than 5. With a second group had a score of 6 for painfulness. Their suffering scores were abnormal, but their disability scores were normal. And then the third group had a painfulness score of 7, abnormal suffering, and abnormal disability. So what that suggests is that there might be 2 thresholds where up to 4 or maybe 5 there is painfulness without suffering, and then above 7 there is not only suffering but also disability. So if we would translate this to tinnitus conceptually, then of course the same might hold for tinnitus. And what we do know is that the correlation between tinnitus and suffering is nonlinear. So that means that up to 4, you will not have, if you have tinnitus on average, of course, a tinnitus of a loudness score of 1 or 2 or 3, or maybe even 4, then that is usually not associated with suffering. But then once you get over 4, there is a threshold above which most people will start to suffer. And hypothetically, once you get over a second score, which could be 6 or 7, then there would also be disability associated with it. And so if you only use linear analysis methods, you will not find a correlation. But if you use nonlinear methods, you will find a correlation with this threshold, with this kind of plateauing. So there is a level below which there is no really increase in suffering, and then all of a sudden there is an increase in suffering. So that's why some people say there is no correlation, and other people say, yes, there is, but it's nonlinear.
I see. Another question. Do you think that targeting this suffering that is caused by tinnitus to be maybe a better option to target rather than targeting the objective tinnitus volume on itself?
Yes and no. So the idea is, well, the suffering network correlates with what we call in cognitive neuroscience the salience network. So if the brain finds that the tinnitus is behaviorally relevant or very distinctive, then it will push it to consciousness. So if you can take away the salience of the, so certainly by reducing the suffering, that might actually also reduce the volume or the loudness of the tinnitus, just like you don't feel your clothes because the clothes are perfectly well predictable by your brain and are not of any salience. Now, theoretically, if your tinnitus does not change, then it should also be non-salient because it's perfectly well predictable and it doesn't have any behavioral relevance. But in reality, that depends to what level you can reduce it, and it might still be there even if you reduce the suffering. On the other hand, if you can reduce the volume to 0, then there should not be, certainly not tinnitus-associated suffering. There still might be suffering, but suffering which is present in every living species in the sense that suffering is non-specific, but you attach it to something. So whether you suffer from pain, whether you suffer from tinnitus, whether you suffer from a divorce, whether you're suffering from losing a family or a friend, the network is very similar. And therefore, this non-specific suffering pathway is going to still be active, but it won't be linked to the tinnitus sound anymore. So therefore, I think that the ultimate goal of research should not be only to look at suffering, but to really try and reduce the loudness, ideally to 0, but certainly to a score below 4, which is livable for most people.
I absolutely agree with you. Now, to share a little bit of my experience, There was a point where my tinnitus was so unbelievably reactive to every single type of noise. It was spiking continuously and constantly. Now, while the amount of sufferers or patients who have this level of severity are maybe fortunately quite small, it does exist. And I think that any sort of therapies that involve the limbic system or the emotional centers of the brain, targeting them would be ineffective for such a level of severity. But I do agree that if the tinnitus does stabilize, which happened in my case, thankfully, then it becomes much easier. Now, a really quick question, the last one. There was a particular large amount of people who were interested in the winding up effect, specifically in delayed or reactive tinnitus, and how it could potentially seem to resemble central sensitization. Do you think that this implies that the mechanisms are more similar than people may believe to be?
Yes, and we've done a fair amount of research to look at the analogy between tinnitus and pain. And if you look at the networks in the brain, they're actually very similar, as well as for some of the pathophysiological mechanisms involved. And so clinically, the winding phenomenon means that if you give certain stimuli with the same intensity or the same amplitude, that normally you will get a very similar response. But if you do that for a long time, or especially in patients who have central sensitization, then the first stimulus will give a certain amount of pain. The second stimulus will give more pain. The third stimulus, even more pain. The fourth, even more. So there is a gradual worsening of the pain, even though the stimulus is exactly the same. So clinically, we call that the winding up phenomenon. And this has been related to a change and gain in the sensory cortex, as you can see on the lower image, because of an adjustment of the amount and sensitivity of the opioid receptors. Now, that being a potential mechanism, the central sensitization is what could be considered the physiological or the pathophysiological cause for this winding up. And this is nothing new. So we looked, based on Agamöller's work in 2000, and Agamöller was my PhD promoter, so I was highly influenced by him. He suggested to me to look deeper into the parallels between pain and tinnitus. And so already in a PowerPoint from 2004, you can see that we were already talking about the wind-up phenomenon, even though we have no clue what it really was caused by, because at that time, neuroinflammation was not yet known. Now, what we did know is that there is different fibers in the nervous system. You've got the small unmyelinated fibers, which are the phylogenetically oldest fibers. So you have them in the lancelet fish, and you have them in the phylogenetically oldest creatures. They only have unmyelinated fibers. And those unmyelinated fibers, which we call C fibers or naked fibers, they basically transmit not the painfulness of a signal, but the unpleasantness or the pleasantness of a stimulus. So that's why they come in two versions. You've got high threshold and low threshold. Low thresholds are the ones that are active when you slowly or gently caress somebody, and the high threshold, or when you inflict pain, then the painfulness is transmitted by the myelinated fibers, so the thicker fibers, but the unpleasantness by the C or the smaller fibers. Now, when you have central sensitization, what does it mean? It means that there is a inflammation. So if you have let's say a cut in the skin, then you'll get a local inflammatory response close to the cut. This will release a lot of chemicals, which we call chemokines, like interleukins and tumor necrosis factor alpha, etc. And this inflammatory response will kindle the C fiber to become very sensitive to extra stimuli. And then if this inflammatory response goes to the dorsal root ganglion, which is still in the peripheral nervous system. We call this peripheral sensitization, and that will cause pain hypersensitivity, meaning that the stimulus that normally causes pain now causes a lot more pain. Now, when this inflammation then spreads to the dorsal horn of the spinal cord, which means the central nervous system, that will induce what we call central sensitization, and that results in allodynia, which means that a normal, non-painful stimulus will be perceived as painful. So that means that if you give a non-painful stimulus sequentially, you will get more and more and more pain because of the central sensitization. Now, when this inflammatory response then spreads to the brain, then we call it centralized pain. And this can spread then to the somatosensory cortex, and that will then result in very frequent attacks of pain. And when it spreads to the medial pathway, the suffering pathway, or the salience network, then that will be associated with anxiety, depression, frustration, etc. So ultimately, sensitization is the consequence of an inflammation that starts at the periphery and then progressively spreads to the brain. So which also means that if you have only a peripheral sensitization, but that does not spread, you might get hypersensitivity. For example, if we translate it now to the auditory system, you might have sound hypersensitivity, which we tend to call hyperacusis. But if you go then to central sensitization, then that might lead to normally painful sounds or sounds that should be perceived as normal, like somebody talking to you, might be perceived as too loud, and actually even being painful if this then becomes linked to the somatosensory system. And if this spreads to the brain, then the tinnitus might become associated with anxiety, depression, etc. So and this sensitization in the auditory system has already been shown, well, at least the inflammatory parameters that are associated with the sensitization have been shown to also exist in the auditory system. So yes, this winding up phenomenon can probably be explained by the fact that you can get an inflammatory response starting in the periphery, so in the cochlea, and then spreading progressively to the brainstem and to the rest of the brain, and progressively adding then more and more stimuli that make the tinnitus more bothersome than just presenting as tinnitus.
Mm-hmm. I'm going to ask a really kind of simple question. Well, it's not. The answer is not simple. The question itself is simple. Many people are curious as to why some people develop loudness hyperacusis to varying degrees. For example, the loudness hyperacusis can become very, very catastrophical. Like, for example, in my case, I could not even drop like a toothpick onto the floor, for example. However, I did not develop pain hyperacusis or Noxacusis, as it's also called. So, well, just generally based on your experience, why do you think some people can develop a hyperacusis to a loudness hyperacusis to a very, very high degree of severity, which I assume is, associated with the central sensitization like you have just described. However, they do not develop Noxacusis. However, some people with loudness hyperacusis being minimal do develop a very, very severe pain hyperacusis or Noxacusis. This has actually also been shown in some research recently that the type 2 afferent fibers, maybe in the cochlea or the outer hair cells, have something to do with this.
Yes. So the hypothesis could be based on what we know from pain, that if you develop an inflammatory response in the periphery, you can get just sound hypersensitivity or sound hyperacusis. However, when it spreads to the dorsal horn, and in the case of the auditory system, of course, to the brainstem, then we know that there is connections also to the somatosensory system. And then, of course, if your somatosensory system also becomes hypersensitive and it starts to become triggered or activated by sounds rather than by somatosensory input, then you can more easily understand why a sound can also cause pain. But that would suggest that you already have connections that are existing. And we know, thanks to the work of Susan Shore, that these connections do exist. But normally, they're not being hypersensitive, except, of course, if this inflammatory soup that surrounds the ventral and the dorsal cochlear nucleus just spreads and also inflames the somatosensory nerve fibers that are there. I'm not saying this is the mechanism, but it's the, let's say, the most theoretically easy to explain mechanism, and that can be tested in animal research. Now, the amount of hypersensitivity could depend of how much inflammatory response there is. Just to put that into perspective, if you are walking and you twist your ankle just a little bit, there might be a small inflammatory response to take care of the damage that's done by twisting your ankle and to heal. Now, if you severely twist your ankle, then of course the damage is a lot bigger, and then you might not even be able to walk on it anymore because of and it's the same stimulus. It's just the stimulus of the pressure on your foot that is tolerable in the small inflammatory response, but is not tolerable in the large response. So also this is testable on the condition, of course, that you can measure the hyperacusis in an animal model, or you can do more invasive, or you can do more local and thus more invasive research in humans. So the problem, however, is that it's not always easy to translate what we know in animals to what we know in humans, because of course you can't ask an animal whether it is hearing tinnitus, or you can't ask an animal whether it's having hyperacusis. You can only infer from the behavior. Yet we know that, for example, if you want to treat tinnitus or hyperacusis in an animal, it will respond to very many drugs, and behavior will be different. And so we say, yeah, we can treat tinnitus with memantine, we can treat it with gabapentin, we can treat it with GABA B ergic drugs like baclofen. And then when we translate that to humans, it doesn't seem to work. And that is because, well, we assume that the animal has tinnitus and hyperacusis, but we don't know. And this is the problem for translating a lot of animal research to humans, where everything seems to work in an animal, which unfortunately often causes a false hope, not only in the people with tinnitus, but also in the researchers, that we finally found something that works. And then when we try to translate it, it does not seem to work. And this is not just a problem in tinnitus research. It's a problem in every kind of research. So this translatability is a huge problem for all research. Now, then you could wonder, why do we even do animal research if we can't translate anything anyway? Well, the reason is that you can translate one thing. Things that are dangerous for animals, like side effects of drugs, or let's say you develop a drug for tinnitus that seems to work in an animal, but it causes severe complications or side effects. Well, you tend to see the same side effects in humans. So animal research is probably more useful for knowing what is a dangerous drug than for knowing whether a drug would work or not. And this is where we have to be always very careful when we interpret animal research that tells us, this is how we think tinnitus works, or hyperacusis works, or Noxacusis works, without human confirmation. in studies that are somewhat similar to the animal research.
Yeah, Dirk, we've seen many times, of course, that research gets hyped based purely on animal results and then later fails in humans. But is the only reason because we can't know if an animal has tinnitus? I mean, could it also be because animals don't have the same higher brain regions as us, and therefore the whole mechanism itself could be different?
Certainly, yes, certainly. If we want to look for animal models of suffering, that is different. Even though every animal has a salience network, the one that is the basic, but maybe the salience network is more related to the arousal associated with the suffering, and how you fill in this arousal would then be more linked to the orbitofrontal cortex, which is more bigger in humans than. But animals do suffer. However, um, the bigger problem in tinnitus is of course, that we don't know whether an animal hears a tinnitus sound or not. But it's not unique to tinnitus. You see the same in pain. You see the same in basically every brain disorder, even in cancer research, where you would think it's more obvious. There is a very, very few treatments that work in animals for cancer that can be translated to humans. So even though we're from an evolutionary point of view, there is a lot of homologies or a lot of similarities between animal brains and human brains. Just like you say, Hazel, a rat is a rat and not a human. And so they do respond differently to different stimuli. Also, their hearing range is very different. So the frequencies that they hear is already different, even though they also have a cochlea, they have an auditory nerve, they have very similar anatomy. It just functions in a different way. So, which means that we cannot infer automatically because something happens in an animal that will happen in a human.
Mhm. Well, what an excellent answer. One thing that I have wondered, well, it's not that I've wondered, but I'd like to hear your take on this. Um, I wanted to hear your thoughts specifically on how inflammation, or more specifically, neuroinflammation, can affect synaptic function. In turn, I guess I would mean oxidative stress as well.
Yeah, so oxidative stress, or basically what we could call, which usually results in excitotoxicity. So what that means is nobody can sprint a marathon. So if you have these C fibers that you see on the screen, they're naked, so they're very sensitive. Now, if you overstimulate them, then you can develop what's called an excitotoxic neuroinflammation, or it's also called neurogenic neuroinflammation. So we know that inflammation usually comes from toxic products. It usually comes from trauma. So it can come from many different things. But what fewer people know is that just a C fiber that is constantly overactive will actually also cause an inflammation. And just to visualize it, if I gently caress your arm, very gently caress your arm for 2 hours in a row, it will become hypersensitive. It will become red. It will start to swell a little bit. It will develop a neuroinflammation just because of, even if it's very gentle, overstimulation. So this neurogenic neuroinflammation can cause a problem. Now, mitochondria, which are the energy factories in our nervous system, when you stimulate them too much, so when you ask the mitochondria to work very hard, and this could be due to being in a very loud, noisy environment where your descending auditory system tries to suppress activity, or there is too much input coming in and those nerve fibers are constantly overactive, that might in itself cause the production of a lot of energy. And one of the side products of energy is these reactive oxygen species. And these ROS or reactive oxygen species actually are normally signal molecules. So they go to the nucleus of the neuron, and they produce they activate then certain genes that will cause anti-inflammatory responses and will cause repair responses, etc. So normally these ROS are beneficial. However, if there is too much of these reactive oxygen species, they become damaging, and they will damage the DNA, and therefore they will then cause a dysfunction of your inflammatory response and a dysfunction of your repair response. And then, of course, you do get that there is no control anymore. And so you get an inflammatory response that is out of balance to the stimulus. And this is just by overactivity of those, especially those C fibers, those naked fibers that basically tell the auditory system that the sound is perceived as unpleasant, meaning that it is too loud, in contrast to activating the low threshold C fibers that then make a sound pleasant, and which is of course why we like music, because of those C fibers, and why we dislike certain sounds like scratching with your fingers on a chalkboard or other unpleasant aspects. So yes, ultimately, overstimulation of those C fibers can cause reactive oxygen species responses that then change the way our neurons will respond to what they think is a perceived trauma that is overstimulation.
Mm-hmm. Okay, now this question has been actually requested a lot. It's not really part of the general questions overall, but there are certain members of the community that believe that drastically changing your diet, or for example, dry fasting, can actually decrease oxidative stress. Many people also refer to this as mitochondrial dysfunction, and potentially fix, so to speak, the tinnitus. Do you believe that could be true?
Yes and no. So it is true that if you conceive of an inflammation as a response that tries to reduce any kind of trauma, whether it is a cut in your skin, whether it is a trauma to the cochlea by too loud noise, whether it is a too much sunshine, any trauma will cause or too much of anything will cause a neuroinflammatory response, which in the acute stage is activating a repair mechanism. Now, of course, the problem is if you already have an inflammatory state, then any stimulus on top of that might actually worsen it. But of course, if you're very sensitive to certain foods, which we tend to call food allergy. A food allergy is basically inducing a neuroinflammatory response to a certain stimulus, meaning a certain food type or food particle. Now, if you already have a pro-inflammatory state, but then more generalized because of these abnormal food particles, and that's what's called the gut-brain axis, then if your microbiome, so your bacteria, but also certain viruses, certain parasites in your intestines, but also on your skin, when they change, then they can release free fatty acids that are also signal molecules that go to the brain and can induce some fear, some anxiety, some neuroinflammatory state. And if you're already in a neuroinflammatory state, when a minor trauma happens, just like when you touch a twisted ankle, then a minor auditory problem might actually, because of this hypersensitivity, might be perceived by the brain as a big danger, and that can of course worsen. So if you then say, well, I'm going to trying to reduce my pro-inflammatory state by adjusting the food intake, then this might work, but only in those people who are hypersensitive to it because of the wrong microbiome or a changed microbiome, or because they have some genetic changes that are sensitive or that make those people sensitive for certain food products. So it will not work in everybody, but anybody who will only eat fat will get some inflammatory states. So, but people who eat a normal food and have no genetic sensitivities might not benefit as much as people who are already allergic or hypersensitive to gluten or to lactose or fructose or any of these types. So in that way, yes, it can. But of course, if you look at that from a scientific point of view, and you mix everybody, you mix the people who have no food sensitivities with the people who have food sensitivities, and you happen to have a larger amount of food sensitive people in your study than non-food sensitive, then your study will be negative. You won't find anything. So first, we need to develop the theoretical concept of, which is what I, for example, just explained, and then that will allow us to design studies that might be positive, even though previous studies were negative, because we just mixed up everybody. And it also means that we should personalize our approaches, and in the future, hopefully, have all this information available so we can tailor the treatment to the genetic polymorphisms or the genetic individuality, as well as the data from the microbiome from the people, as well as having all these chemokines that you see here on the slide, have them and see, okay, well, there is a clear inflammatory state, so it makes sense, and the people have some genetic polymorphism, so it makes sense to put them on a diet. Whereas if you put everybody you see on a diet, well, it won't work for many of them because they might not be the right patients for it.
So basically, overall, something like, I guess you could say, dry fasting or a carnivore diet, which has been recommended by certain members of the community, which, by the way, I think is a bit counterintuitive because it has been associated with increased levels of pro-inflammatory markers, would not be a good idea for absolutely everybody.
No. Just like there is no single treatment that will work for everybody. We were hoping that there is a final common pathway that will work for everybody. But telling everybody to go on a diet, telling everybody to start eating meat because it has some more tyrosine in it, which is a building block of our dopamine system and noradrenaline, or and because it has the amino acids that we need to develop signal molecules, does not make sense. But we have the tools to look at many of these aspects in people to then tailor the treatment to the patient. However, this comes at a big cost, and it's not always easy. Like, for example, if you want to look at these chemokines, then you need to take it, put it immediately on ice -40, and then have it analyzed. And there is some variability in these results. So we're still not ready yet to really use these at an individual level, but that's just a matter of time. And then we can tailor and tell who might benefit from an anti-inflammatory diet to people who might not benefit from anti-inflammatory. So if you just look at this, at what I've mentioned, then you could imagine that, for example, whether you have hyperacusis or not might already give you some hint, because the idea is that the hyperacusis might actually, yes, be related to a peripheral neuroinflammatory state, and maybe even in the dorsal horn, and that they might potentially more benefit from food supplements or a diet that reduces TNF alpha or interleukin 1b that you can see here in those slides, and that might increase interleukin 10. So if we know which foods and which food supplements do that, then if you have hyperacusis, then it might make more sense to use it than if you have no hyperacusis, also just from a theoretical perspective.
Okay. And I know we should be moving on here, but I have one last question about diet. Now, something that has actually shown to help some people with their middle ear issues, specifically with some forms of Noxacusis, is a low histamine diet. And it has actually not shown the same efficacy as for example, taking antihistamines. What do you think that could be?
Well, histamine is an arousal neurotransmitter. So it's a neurotransmitter that is used in the nervous system to create arousal. Now, antihistamines block histamine on the receptors, whereas a low histamine diet still has histamine in it, just a lower level. So it's different whether you block something or whether you're exposed to less of it. For example, blocking something is like if you're in the sun and you use a sun protection factor number 50, then you're fairly safe in the sun. But even if you have a very sensitive skin, let's say you are an albino, then even if you go in the sun but expose yourself to a lower level of sun, you will still get burned. So it really depends on your individual sensitivity state as well as how you block it. And so blocking is usually more, blocking the receptors like by using antihistamines is probably more beneficial to having a lower amount of histamine, because we know, for example, that blocking your histamine will improve your REM sleep and your deep sleep. And recent work of Robin Guillard and friends has shown that the amount of deep sleep and the amount of REM sleep actually correlates to the loudness the perception of those people who are sensitive to sleep, determining the loudness. And that seems to be related to the amount. And you could imagine that even with a low diet, that might not have as a powerful effect on the amount of deep sleep and the amount of REM sleep as antihistamines have. So there might be multiple ways why there is a difference.
Excellent. Okay, well, I guess let's go ahead and move on.
Yes. So the some important questions relate to tinnitus ontology and subtypes. So, and this was based on a study published in probably the most important medical journal called the New England Journal of Medicine, where they described ontologies as systematic representations of knowledge that can be used to integrate and analyze large amounts of heterogeneous data, allowing precise classification of a patient. So, and the question then, so basically what does it mean? It means, can we find subtypes of tinnitus that we can then tailor our treatment to? And would that improve the way we respond? So in that same article, they looked at two people who present with the same genetic deficit. Yet if you look at one is very petite and the other one is a tall person, and they have different symptoms of a whole branch of symptoms, but they really present what we call phenotypically, so in the clinic, with different complaints. There's few overlapping. There's like 3 overlapping symptoms, but then there is 3 or 4 different symptoms. Yet genetically they have the same problem. And this is where a problem arises, that just using clinical features, we cannot create classifications. And it's not that we haven't tried. I mean, looking at the TRI database, looking at other databases, people have used the same clustering analysis that we use to look at painfulness with suffering and with disability, or without suffering and disability, but we cannot find any subtypes, even though we use databases of more than 4,000 people in it. And that's exactly what this paper in the New England Journal of Medicine was showing, that yes, there is a difference in the genetic expression and the genetic features. So what do we have to do? Well, we have to look at way more features. So in tinnitus, that would mean if we try to classify based on the clinical presentation, whether it's left side, right side, pure tone, noise, even with or without hyperacusis, whether without Noxacusis, we cannot create subtypes. What we need to do is we need to add extra information. So for example, genetic information, as in the example given here, but also maybe minimal masking levels, audiometries, every amount of information that we can get. And then we might be able to find subtypes. The problem is, in tinnitus, these databases don't exist yet. So we still, it would require, and this would be a beautiful large European or a large American grant, where we select from many different countries as much data as we can, and this is and then use more complex artificial intelligence than what we're currently using. Currently, we're predominantly using machine learning and maybe some deep learning, but no foundation models yet. No models like ChatGPT that's basically integrating very heterogeneous amounts of information and then is trying to generalize very, very heterogeneous amount of information. Now, what we do is we say, okay, here is 1,000 EEGs with tinnitus and 500 without. Show me the differences, and then maybe we look. But that's based also, again, on one feature, on brain activity. Well, maybe we have to add audiometries to it. Maybe we have to add questionnaires to it. We have to add tinnitus measures to it. So, and that will allow us to create subtypes that we might then be able to group and find treatments for those subgroups. But of course, from a theoretical perspective, you can go a step further and say, okay, let's say in pain, we know there are 7 subtypes based on the ICD classification. Now, let's say we find 7 subtypes of tinnitus, but then within the subtypes, there might even be subtypes, and ultimately you end with n equals 1 with the individual. And so ultimately, but in order to be able to treat the individual, we need yet a lot larger databases. So the current problem is we don't have those very heterogeneous databases that give us lots of information, and I mean databases that are large enough. We have them for clinical features. We're currently having them for EEG, even though they're very rare. And we have them for EEG and audiometry, so we will get there. But we're only at the starting phase of this. And of course, then, as a tinnitus researcher, we don't understand how these foundation models work, or how this deep learning works. And that's where it's very important that you collaborate with people who are very knowledgeable, like computer scientists or big data people. But they have to understand what to put in their models. So it has to be a collaboration between clinicians and computer scientists and statisticians, etc., in order to develop those subtype specific treatments.
Yeah. Dirk, I have sorry, Anthony, if you don't mind, because as a very key question that comes to mind for me, I would love to see such a big data project happen, by the way. I really would. And I think we could also leverage places like Tinnitus Talk for things like that, at least for the online data collection part. And I'd love to work on something like that. But I'm also thinking, like, if we have limited funding, which we do, right, should we invest in a huge, big, costly data project to figure out what the subtypes are first and then move to the treatments? Or should we skip the subtyping part and just move directly to the treatments and then through that figure out, because you're going to anyway, once you start testing out different treatments, also figure out like what works for whom, and you're going to start fine-tuning the treatments and figuring out this works for this subgroup, this works for that subgroup, etc. In the context of limited funding, what do you think is the best
I'm
approach?
sorry to interrupt. Before Dirk answers, I would like to add something to Hazel's question as well. One thing that really worries me is the prevalence of self-reporting bias in these types of big data projects. I'd like to add, like, what can be done to I guess, to avoid this, especially when it comes to such subjective conditions.
Yes. Well, to first answer Hazel's question, I don't think that private research group like Tinnitus Quest should fund these big projects because, first of all, they require a huge amount of money. And that's where we have the European community for, and why we have the American and then the Chinese. And these should be very large international, well, in the European context, of course, multi-country approaches that should be funded by the European community or big country funds. And probably a grant or an agency such as Tinnitus Quest, who wants to create funds, can in parallel already try and look for treatments that might even benefit a smaller group, but based on some mechanism-based approach. So if we know a mechanism in a group of people, then we can say, okay, if this is the mechanism, then we should be able to try and use this mechanism in order to treat it. For example, when people realized that diabetes was related to insulin, then they said, okay, let's just give those people insulin because they have not enough insulin production. So we just give them insulin, and indeed the diabetes improved. So this was a purely mechanism-based treatment. There has never been one placebo-controlled randomized trial to show that insulin works better than no insulin. Just like in the British Medical Journal, there was a paper published that said, well, there has never been a study, placebo-controlled, randomized study that shows that jumping out of a plane with a parachute is actually safer than not jumping out of a parachute without a parachute. And of course, this was meant partially humoristically, but it also said that if you have a very solid mechanistic approach, well, then maybe it might be more ethical not to require a placebo-controlled trial if that is going to endanger the people, and we can just do if we can then show that indeed this mechanism is normalized after the treatment, and this normalization is associated with a clinical improvement, wow, do we still need placebo-controlled trials? And of course, this is where I think with less money, you can make a huge difference. So if we focus on what part of the tinnitus mechanism we understand, then it makes sense to do a trial. And if we see, yeah, well, there is some benefit, but not enough, we can look at what part of the mechanism that we not address in the correct way. And this is research that can go in parallel with the larger subtypes, with the larger studies that should be funded by national granting agencies, or even international in the European sense. And of course, a large study comes with a reporting bias, as was mentioned, and this is there is not just the reporting bias. There is a lot of biases that are at stake, and that's why it's very important that researchers always make clear whether there is a reason for them to have a bias. If I have a company and I'm selling a drug that might work for tinnitus, then of course I'm going to be biased. Whether I want it or not, I will still be biased in how I interpret the results of the study. If I believe in something, I strongly believe in something, then I might also be biased in how I look at the data and ignore a little bit the things that don't fit with my belief. And so there is a lot of biases in not only reporting, but also in analyzing the data. And of course, the more people that are involved, the more different kinds of biases you will have. And this makes it sometimes a little bit difficult in studies that are multi-center, because it also depends on what type of people you see. For example, it took me a long time to realize that being a neurosurgeon, I see different patients than if you're an ENT specialist, or certainly than if you're an audiologist. And so the way I look at a tinnitus, at a person with tinnitus, is very different than an audiologist. And this will also result in the longer term that I see way more people with a grade 3 or 4, so very severe tinnitus, than if I would be an audiologist. And therefore, if I report something, it will be biased by the kind of people that I see. And an audiologist will be biased, and a psychologist will be biased, because people come to me as a neurosurgeon, being a car mechanic or an electrician, and looking at it from a very mechanistic point of view, whereas a psychologist might be not very mechanistic. And so you already attract a different types of people, and therefore what you write is what you write, and what you publish and what you see is biased. And whether you want it or not, even if you don't know, you're still biased in the way you report it and the way you look at it.
Okay, I think that's an excellent explanation. Yeah, I absolutely agree with you. The bias is absolutely immense, and we see it very often, especially with, for example, certain types of treatments that are being offered to certain types of subgroups of tinnitus sufferers. There is a tendency that we can see with some audiologists, offering certain treatments and then just outright denying people who might be like, for example, of the reactive tinnitus subtype or the Noxacusis subtype, and selectively choosing just the people who they know will react well to the treatment, whether that be placebo or if the treatment is actually working. And that is a major issue, and it can lead, of course, to disastrous consequences. Okay, I guess.
Well, yes, and I give you a very other practical example. I was having a discussion with the CEO of Neuromod, and he said, well, you see a lot of patients. Why do you not use Neuromod that often? And I said, well, I think because we see a different group of people, and we are predominantly interested in neuromodulation. But he said, yeah, but our tongue stimulation is also neuromodulation. I said, I agree. But we have already for more than 20 years been doing transcranial magnetic stimulation, transcranial electrical stimulation, neurofeedback, and all these other neuromodulation approaches. So we know more or less who might respond to what. We can't predict it at an individual, but we know more or less who might respond. And then, of course, the new kid on the block, being it tongue stimulation or vagus nerve stimulation, always gets selected to be the last one to try. Well, if nothing else helps, we can try this. But the problem, however, is that we are biased, or we select already on who does not respond to neuromodulation, whether it's TMS, TDCS, etc. Well, they're very unlikely to respond to the tongue stimulation either, because it's just another form of neuromodulation. And that's why when we looked at our data, people were seen not to be as responsive as in the studies. But that's because we selected already people on beforehand who did not respond to any kind of neuromodulation, so they were very unlikely to go. And so this is indeed very biased. So if we would publish our data, then of course it's extremely important that we explain or try to explain why we think that our results are not as good as somebody who's naive to any kind of neuromodulation, who has never had it. So if we would use it as the first kind of treatment, well, our success rate would be very different than it is currently. And this is exactly what you mentioned. Yes, everybody is biased in their results because of their selection criteria, because of the type of people that they see, and because of their own beliefs in what they present first and what they present last.
Okay, excellent. I guess let's go ahead and move on.
So there's also a lot of interest in somatic tinnitus and the influence of the trigeminal nerve. And one of the questions is, why has Susan Shore's important research on trigeminal involvement in tinnitus been confined to specialists in orofacial pain and chiropractors? Also, where does somatic tinnitus influence fit into the triple network model, and why does the clinical tinnitus community not prioritize somatic influence in research and practice? Well, the third question is the most easy one. That's the first slide I showed. It's all about how much money is available. And second, there is, however, also a scientific reason. The prevalence of somatic influences in tinnitus is big, with numbers going from 30 to 70% of people having an influence of their tinnitus by somatic maneuvers. So there is a group of tinnitus researchers who say, well, if 70% of the people have it, well, we shouldn't really look at it as a subtype. It's actually the most dominant type of tinnitus. So we are not that especially interested in studying the specific somatic influences, because it's the norm. Of course, if in your research center you only see 30%, then of course it makes sense to look at it from a niche or a subgroup point of view. So this is probably one of the reasons why people do not prioritize the somatic influence in research and practice. Also, if you see somebody with somatic modulation, not always, but most commonly, it's a worsening, whether it is when you clench your teeth or whether you move your neck, etc. So then the idea is, well, if we can reduce that, we can maybe stabilize the tinnitus, so we can remove the modulation of it or the fluctuations, but ultimately we're still having tinnitus. So that might also be one of the reasons why it might not be as attractive to address as something that you hope could really reduce the sound and therefore also the somatic modulation. Now, I don't think in general that Susan Shore's research on the trigeminal involvement has only been confined to orofacial pain specialists or chiropractors. The problem, however, was that there is no tool that we can use in clinical practice currently to treat somebody. So I know she has a research device that might become commercialized, and then of course many more people will want to be using it depending on the outcome of the studies. But also a second reason, apart from the fact that there is no tool that we can use, is that Susan Shore's important research is difficult for clinicians to understand. All clinicians understand the concept that there is a modulation of the somatosensory system on auditory information processing, not only in the dorsal-cochlear nucleus, but all the way to the auditory cortex, and so that they influence each other. Yet the details are, even though they're very eloquently detailed by Susan Shore's group, is difficult to use in clinical practice. And so I remember when she first started with it, I visited her lab and we had a very nice time together trying to understand this. And so I said, but you have to see, Susan, I'm a very simple clinician. I want to use this. And I said, so if I understand you well, then the somatosensory influence on the auditory neuron is mostly inhibitory. Yes, it says most is inhibitory, but there can be activating. And I said, yeah, but I don't want to know all of that because I can't use it. But my simple assumption is if I stimulate, and it's mostly going to be inhibitory, it might inhibit the tinnitus because it might inhibit the tinnitus. And that's the way how clinicians look at it. And of course, that is very different from the way Susan Shorter looks at that. But we went ahead and we tried C2 stimulation, but not timing it at any way. I just looked at the numbers and we saw that we did have some people where it really worked, but out of 300, there was only 6 patients where it completely took the tinnitus away. And we implanted them with an electrode, a subcutaneous electrode, in those 6 patients, but we had to select 300 to find those. And that is, of course, because we didn't really understand Susan's work. And at that time, it was very limited. There was very limited knowledge. That was before she really did a lot of her research. But so clinicians want simple principles. And I think her work is very beautiful, but probably a little bit too complex for a clinician to easily translate into the clinic. Now, the second question, where does the somatic influence fit into the triple network model? Well, the triple network model is a theoretical model that was developed by Menon to try and explain cognitive changes in mental disorders by saying, well, there is 2 cognitive networks. One relates to the external world, one relates to the internal world. Or to put it simple, one is about thinking without an external trigger, and one is solving a problem or thinking about an external trigger. And so if nothing goes on in the environment that demands your attention, you start mind wandering, etc. That's your default mode that's active. Then if somebody calls your name or somebody calls fire, then there is a third network, which is a salience network that says there's something behaviorally relevant. You have to respond. That will shut down the default mode network and will activate your externally oriented cognitive network, which we call the central executive network or the frontal parietal control network. And so the salience network determines who of the 2 cognitive networks becomes dominant. Now, part of the default mode network, so the network that says it's not important, there's nothing important, so I can mind wander, I can think about the past and the future, involves the pregenital anterior cingulate cortex and the ventromedial prefrontal cortex, so a part and the midline of the frontal cortex, and that normally suppresses input from the outside because there is nothing important in the outside anyway. So, and it also, next to the sound suppression network, there is also a somatosensory suppressing network that goes from the same area and suppresses then the sensory input because there is nothing important. And when the brain picks up something important, then this suppression will stop. So, of course, the somatic influence requires really that you then integrate the somatosensory and the auditory suppression network because they run in parallel, so they can influence each other very easily, especially at the level of the periaqueductal gray, but also further down the pathway. So the somatic influence needs to be seen in the concept of the balancing act of sound and pain suppression with pain provoking stimuli and tinnitus or sound provoking stimuli. And the mechanisms are very similar, and the only thing you need is a connection between the two. And therefore, this has been shown by Susan Shore. So basically, it extends the sound percept to how you deal with the sound percept, whether you will suffer from it or whether you will not suffer from it.
Mm-hmm. I think that is an excellent answer. I absolutely agree with you that clinicians or this type of research that, I myself kind of dove into very deeply is definitely above the normal threshold of what a clinician should know. And unfortunately, the somatosensory or the excitatory somatosensory connections to the dorsal cochlear nucleus through carpial cells and fusiform cells is definitely not understood by many clinicians. And I really hope in the future that, maybe when the Susan Shore device does come out, that this overall understanding of this, in my opinion, very, very important connection is brought to light or realized by most of them. Okay.
Yes, but you have to realize that a clinician is like me driving a car. I know how to drive a car. I have no clue how the car works. So if something breaks down, I can't fix it myself. I have to go to a garage to have it fixed. But I can drive the car. And this is how a clinician thinks about pathophysiology in general. They know how, in general, more or less how to deal with it if it's built for them. So if Susan Shore, with help of engineers, etc., builds a device, then the clinician will learn how to drive the device, just like they learn how to drive a car. And some people, of course, also understand how the car works. And when something breaks down or when something does not work, they can know how to fix it. And this is where some specialists, but certainly not everybody, some clinicians will learn more in detail about Susan Shore's how the device works and on what principles, so that if they have not the result that they hoped for, by rationalizing about it, they can come up with a fix of why it does not work. Whereas the person who knows how to drive a car, but if it doesn't work, well, can't solve it, they will just acknowledge, like, look, it doesn't work, and everybody will have to accept it. And so you will always have two kinds of clinicians. And I don't think that it is to be expected that all clinicians will understand the mechanism behind the device, but they will learn how to drive it.
Excellent analogy. Very good. Okay, I guess let's move on.
So then here is some of the studies, and why these are the two more recent studies, and why is it important to try and read studies is because it helps you to put things into perspective. We can start with Susan Shore's device or the tongue stimulation. Doesn't really matter. They both have their strengths and both have their weaknesses. First of all, Susan Shore's work, but also the tongue stimulation, is based on animal studies and specific mechanisms that really predict what you should do, but more even for Susan Shore's device, because the timing is extremely important. That if you have this spike timing dependent plasticity, then you have to combine the sound and the electrical stimulation in a specific way. Now, if this would be the common mechanism for everybody with somatosensory modulated tinnitus, then you would expect very robust and very strong results. However, if you look at the data with an intention to treat, which means that you look at the analysis of everybody, whether they've finished the treatment or not, and the assumption is that most of the people, not all, but most of the people in the trial will actually stop because they don't see a benefit. So you should kind of count them to the negative results and therefore also include the negative results. So that's the purpose of the intention to treat, rather than the treatment. For example, if you start with 100 patients and only 14 finish, and all 14 do well, then you can say this is a brilliant treatment. Yeah, but only for 14 patients, because there was 86 who stopped because they might not have had a benefit result. So that's the goal of the intention to treat. Now, if you look at, you can look at loudness and you can look at suffering. So if you look at the loudness results, you see that there is a benefit in the green dots that is clearly more than in the control group who had only auditory stimulation. Now, if you look, the problem here is that it measures the changes in loudness in decibels. So it's a minimum masking level, and this does not correlate with subjectively perceived loudness. So for a clinician, that is difficult, because they say, well, what does it tell me? I can't tell my patient that the tinnitus will sound 10 decibels less loud, because that does not correlate with the subjective loudness level. Now, this is not Susan Shore's fault. This is the fault of the FDA, who required her to take these outcome measures. And this is, of course, driven by audiologists, maybe academic audiologists, or people who like numbers that can be repeatable. But that's not always the clinically best. And we looked at that from the brain point of view, and the minimum masking level has no neural correlate whatsoever, whereas the subjective loudness perception, like if you ask on a scale from 0 to 10 or from 0 to 100, how loud do you perceive it, does correlate with certain brain activity. Now, so the loudness measure is unfortunately not the most optimal for a clinician. Maybe for a scientist it is, but not for a clinician. Now, if you look at the subjective improvement, then what you see is that the benefit on the TFI and the THI, which are two questionnaires that look at the suffering or the distress associated with tinnitus, then you see a benefit of minus 12 points on the TFI and minus 5 points on the THI. Now, the way that scientists try to figure out, well, is this clinically beneficial or not, is by using something that's called the MCID, which is the minimally clinically important difference. But you can compute that in 4 different ways from a computational point of view, and they all give different results. Now, the MCID, so the minimum benefit you should have to be relevant is 13 points, and Susan Shore's device has a benefit of 12 points, which is just at the edge of being clinically relevant, but only minimally clinically relevant according to the definition. The THI is the same, 5 points of benefit on the intention to treat versus an MCID of minus 7. So what does it tell us? Well, it tells us that these scores, or scores out of 100, now that means you got about between 5 and 10% benefit. Now, if I'm a patient and I suffer, just to put it in simple figures, and my suffering score is 90, and I go to 78, I'm still suffering severely. So this is not something that, and these are averages, of course, but this is not something that you think, okay, this is going to be the big breakthrough where everybody goes from 90 to 30. No, it gives more or less a 10% benefit. And this is the same benefit that you see from cognitive behavioral therapy, the same benefit that you see from acupuncture, the same benefit that you see from TMS, the same benefit that you see from TTCS. So whatever we do, we seem to arrive at about 10% benefit in suffering scores. And this is clearly not enough. It might be enough if you have a score of 5 and you can drop to a score of close to 4, then you might just hit the threshold for suffering and not suffering. So for a small subgroup of patients, because of this nonlinear correlation, it might be beneficial. Now, if you look at the tongue and auditory stimulation, then these results are very intriguing, because they compare the pairing of sounds and tongue stimulation, and irrespective of what you do, you get a benefit, which is 13 points. But there is no difference whether you pair it or you don't pair it. It seems that if you modify the somatosensory cortex and the auditory cortex, you get a benefit. And this is why they had to do a study for the FDA to get accepted in the US, where they first had to do an auditory stimulus, see what benefit there was, and then add the tongue stimulation afterwards. And they needed to have clear superiority of the combined stimulation versus only the tongue stimulation. And because they had this benefit, irrespective of whether it followed the mechanism, because if it's mechanism based, you would say only with the specific pairing it should be better, but it was not. It was better with whatever way sound and the sound and the electrical stimulation of the tongue was spared. So, of course, the tongue stimulates the trigeminal nerve, whereas Susan Shores stimulates the C2 nerve. But these two nerves do get into the brainstem, and they extend into the spinal cord to the level of C2. They form one big nucleus. So the trigeminal and the C2 nerve are integrated at the level of the brainstem. So it's not surprising that both seem to have some benefit. Now, I just added this slide to show that depending on which questionnaire you use, that you might have different outcomes, because as you've seen in Susan Shores, for one score, she had only 5 points of benefit, whereas for the other, she had 12. How can you explain that? Well, that is because all these questionnaires have a little bit of a different grouping of questions. So, for example, if you look at a tinnitus reaction questionnaire, then the tinnitus reaction questionnaire actually has 0 questions on specific items for tinnitus, and has 0 questions regarding auditory perception. So it is really only looking at a psychological response and on the impact of lifestyle. So this is a very kind of an unbalanced questionnaire, whereas if you look at the opposite, which would be the tinnitus functional index, there you have the most evenly distributed questions, where the amount of questions related to specific tinnitus effects in orange is present. There is a similar amount of questions related to auditory perception. There is questions related to sleep. And there is, of course, questions related to the psychological and emotional impact. The second most balanced would be the tinnitus questionnaire, the tinnitus handicap inventory. Basically, the THI, as you can see, the tinnitus handicap inventory has no questions that relate specifically to tinnitus, and only 4% that relate to the auditory impact. So it's not a very auditory focused questionnaire. It doesn't mean it's not a good questionnaire. It's just not very auditory focused. And that's why if there is more questions related to tinnitus, that you could have a different score on the tinnitus TFI, on the tinnitus functional index, versus the tinnitus handicap inventory. So you should not be surprised by it. Now, this is what I mentioned, just to talk a little bit about what is suffering and what is loudness perception. If you have an auditory stimulus, but it's the same whether you have a visual stimulus or a somatosensory stimulus, your brain will respond in 3 ways. One is a cognitive response, which we've just explained, the internal and external response. One is the emotional response, and the third is the autonomic response. The cognitive response, we can try and use specific questionnaires for it. Same for the emotional response, and for the autonomic response, we can also do, for example, you can use the DAS questionnaire, or you can also measure it by looking at pupillometry if you want to use some more a tool to detect it, or heart rate variability, or skin conduction tests. So the autonomic response you can collect in more different ways, also physiological ones. Whereas the cognitive and the emotional response tend to be by questionnaires or numeric rating scales, where you ask somebody from 0 to 10, how unpleasant is this, how much frustration, etc. Does that cause? Now, suffering means that the sound that you perceive, whether it's an external sound, as in music, or in another sound that you present, or whether it's internally generated in the brain, if the cognitive response is negative, if the emotional response is negative, and if the autonomic response is sympathetic, meaning there is arousal and valence attached to it, and these are negative, then we call that suffering. So this is a very physiology-based definition of suffering, meaning that suffering is a negative cognitive, emotional, and sympathetic response to a stimulus. And of course, you might say, well, so what does it mean? Well, it means that, for example, in pain, of course, most people dislike pain. And the cognitive and emotional response will be negative. But my son, who does judo, when he does not feel pain in the evening, he says, I didn't train hard enough. So for him, the pain is actually a positive sign. So he's not suffering from the pain. For him, pain is just the fact, yeah, I did a good training. Some people in the somatosensory domain might even enjoy pain, like in certain sadomasochistic sects. They're not suffering, but it's very context dependent. And so the suffering is really when there is a negative cognitive and emotional response. Also, it means that suffering is not only emotional. Suffering has a large cognitive component to it and an autonomic component, according to this definition, of course. When the suffering becomes and the sound becomes chronic, your brain will say, as from now, I consider the sound part of who I am, the self. And that's one of the reasons why, from that moment on, your brain will consider yourself as the self plus the sound. And this makes it just more difficult to treat if you have it for a longer time. Now, the suffering self then might become, that might then lead to disability or decrease in quality of life, for which there is still other questionnaires, and might also cause sleep problems, for which there is other questionnaires. So it will have a functional and social impact. And so this is a theoretical model of how we can try to separate different questionnaires and subgroups, but we have not, unlike pain, yet found the thresholds, which I assume would be very similar to when every sound becomes bothersome to almost everybody, and when every sound becomes even disabling. These are average, of course. They might differ from person to person, but this is something that we still have to figure out. And I assume it will exist because it also exists in pain.
Okay, now I probably should have asked this earlier, but I didn't want to interrupt. Now I'm going to talk actually a little bit about Neuromod's study and also Susan Shore's study. Now, a major problem that I think Neuromod's study on Lenire actually has is, of course, the lack of placebo control. And you might think that initially speaking, it's not an important thing because, well, you have this comparison of the auditory only and these, combined with the somatosensory stimulation to the tongue, which I believe engages mostly the hypoglossal nerve or the 12th cranial nerve. There is some trigeminal involvement as well. But the major issue is that Lenire was released in Europe many years before it attempted, or Neuromod attempted, to have it released in the United States. And everybody in the tinnitus community knew about it. I mean, it was all over the news. It was like just everywhere. Yes, and so this, in my opinion, this increases the necessity of a placebo control group even further, because I would assume that most people who are participating in this trial already know what it is. They know that it's supposed to be with the tongue stimulation and the auditory stimulation at the same time. And if you lack a placebo control, or for example, like in Susan Shore's study, the double-blinded placebo control, then how are you supposed to understand this data? I mean, with the lack of this group, you might as well assume that the group that was receiving the combined stimulation was just experiencing more of a placebo effect.
Yes, that could be correct. But if you look at their outcome, their THI score is a lot better than the one from Susan Shore. And this might be related to the placebo effect. That's correct. So I'm not, I fully agree. I just don't know how you would do placebo tongue stimulation. Like, it's also very difficult to do C2 placebo stimulation, because even subthreshold stimulation for sensory feeling, so for not having paresthesias, has some effect. So I'm not sure how you would do placebo stimulation in the Lenire device. I mean, the auditory, you can't, because you hear it or you don't hear it. And the tongue stimulation is very difficult as well, because you could say, well, the stimulator is on, but there is you won't feel any paresthesias. Well, that will create a bias as well, because they say, well, I think now I'm in the control group because there is no tongue stimulation. So it's from a technical, I fully agree with you, but from a technical perspective, it's not easy to do. And this is something that, you really have to try and be very pragmatic in those things. And then you say, well, what's second best? Well, second best is to either pair it or unpair it. But then, of course, you could expect, or you might expect, that a grant agency, the FDA or the CE would say, well, look, there is no proof that the pairing is essential, because all groups, the paired and the unpaired, actually improved. So we won't grant you the status that it is FDA approved or CE approved. But then I think the agencies then decided, look, it is safe. It seems to improve around 13% of it gives a benefit of about 13%. It's not dramatic, but it's in keeping with what we see with other neuromodulation devices like TMS and TDCS and acupuncture. So let's just give it its approval, because it doesn't seem to be dangerous, and it seems to be doing the same thing as other neuromodulation devices. And again, that's probably because they realized that doing a pure 100% proof placebo stimulation might be very difficult to do.
Well, to answer that question, actually, and by the way, this is something that is very niche. Many people don't study this for some reason, at least specifically in tinnitus. The principles of spike timing dependent plasticity are fully or solely based on the timing of the postsynaptic spike in action potential relative to the presynaptic spike in action potential. So to answer the question about how a placebo control might be introduced, well, you just introduced the wrong timing. So as it was done, for example, in Susan Shore's research, or Susan Shore's study more specifically, I think it would have been absolutely possible, I think, for Neuromod to go ahead and do this, because the very specific timing that is required for induction of long-term synaptic depression is very specific. So we're not talking hundreds of milliseconds. We're talking like milliseconds. Yes, and depending on the pairing of the stimulation of the somatosensory system, together with the fusiform cells, or the targeted frequency of the fusiform cells, you can actually achieve basically either long-term depression, which happens in the active treatment group in Susan Shore's research, for example, While you wouldn't want to induce long-term potentiation to worsen people on purpose, that would be terrible. But you can actually widen the timing enough. For example, I'm not going to go into the details because it's very specific, but you can widen the timing enough that there is actually no benefit to this bisensory stimulation. So what I think about Neuromod now, I think that they had a very targeted approach towards FDA approval. And I think that they passed the FDA approval process with flying colors, obviously. They assumed that it was good enough. But now this is not based on clinical data whatsoever. This is based solely on the data that comes in from the community. So I understand that some data that comes from the community is purely anecdotal and not really taken seriously, maybe in a research perspective. But we have actually seen numerous people worsen from the usage of the Lenire device and also see no benefit as well. And it's unfortunate, we really wanted this device to work, but based on the principles of how spike timing dependent plasticity works, just how the Lenire device is set up in general does not promote long-term depression. It can definitely, but it is much less prevalent because as the Lenire device works, it has the Bluetooth headphones that play a continuous maybe preset of audio. And so, of course, this leads to, different type of audio. There's like white noise, et cetera. And in relation to the Susan Shore device, which provides millisecond targeted frequency stimulation, which then in turn is also coupled with the somatosensory stimulation separately. It kind of, I guess, creates these conditions for long-term depression much better than the constant stream of noise that comes from the Lenire device. So in other words, and not to mention the Lenire device does not target the specific frequencies of the maybe fusiform pathways that are hyperactive as well, as it's just general presets, for example. And, basically my question to the Neuromod team is, how is spike timing dependent plasticity, specifically long-term depression, supposed to be initiated if you have a constant stream of action potentials flowing into the auditory system, coupled with the somatosensory stimulation to the tongue, which is, as I mentioned before, 12th cranial nerve, trigeminal nerve as well. It seems from a scientific perspective that it would be impossible to do. Achieving this long-term synaptic depression. So that's just my quick commentary on how I guess like in a for example, spike timing dependent plasticity based device, a placebo group could be actually organized. Okay, well,
yes. I agree with you, and this is actually what they tried. I think they tried to pair it and unpair it, but it didn't really make a difference. Now, I already from the beginning, because I strongly believe in the Bayesian brain, which means that your brain constantly makes predictions about the future, and then uses your senses, auditory, somatosensory, visual, etc., to prove or disprove your predictions based on a model in your head that you have about the world and yourself and the world. So from a Bayesian perspective, of course, if you stimulate your tongue, it predicts much of the time that food is going to come. Now, if food will arrive in your mouth, it will activate your vagal nerve. And so what they might do is they might pair non-tinnitus sounds to vagal nerve, or they might actually also basically activate the vagal nerve, and the vagal nerve is known to reduce anxiety, is known to reduce depression, because the vagal nerve is normally activated when there is nothing of importance in the outside world. So my hypothesis from the beginning was that this was more a vagal effect than a really spike timing dependent plasticity effect. And this is actually shown also by their studies where they pair it or whether it's strictly paired or not paired, doesn't really seem to matter all that much. But what is common is that they activate the vagal nerve indirectly, but very much in keeping with the Bayesian brain. So that being said, maybe their mechanism of action is very different from Susan Shore's device, and therefore we might not really be able to compare them from a physiological point of view. And my hypothesis is actually relatively easy to test. I mean, you could do the stimulation, record heart rate variabilities that associated with, and then see if the change in heart rate variability correlates better to the improvement in the tinnitus distress than for example, the pairing or the unpairing. So, and that might be just another explanation. So, and this is where sometimes it is important for basic researchers to also consider alternative theories. And this is known if you look, for example, at paleobiology, which is very different than what we're talking about now. But you have three big researchers in the field. You've got Simon Conway Morris from Cambridge. You've had Richard Dawkins from Oxford, and you had Stephen Jay Gould from Harvard. I mean, all three really big shots. They looked at the same stratification data from from the same fossils, basically, and they came up with three different theories. One said it's a very gradual, clearly Darwinian evolution, which was Richard Dawkins, and that's because he was Darwin's gladiator, basically. And then you had Stephen Jay Gould, who was actually a Marxist, but he believed in Hegelian philosophy. And here we come again to the word of bias, because he was an Hegelian philosophy basically means you've got a thesis, an antithesis that says the opposite, and then a synthesis that's a mixture of the two. And then you get not gradual, but you get saltatory, you get jumps in evolution. And he used this to explain exactly the same data. And then Simon Conway Morris, who was a Christian who strongly believed in the works of Pierre Teilhard de Chardin, who came up with the idea that in the beginning, there was different species, but we all will evolve to one single species, which will be godlike, and that's the omega. And so he came up where Stephen Jay Gould and Richard Dawkins said, okay, well, we've got a diversifying evolution. Simon Conway Morris, based on the same data, said, no, we're actually converging. We have a convergent evolution. So here again, understanding that there might be different mechanisms involved in the clinical results is important, because maybe tongue and auditory stimulation is based on an entirely different model than the one that they posit. And a beautiful example of that is where Thales of Mileto in 585 before Christ perfectly well predicted the solar eclipse, but he used the wrong model. He still used a geocentric model and not a heliocentric model. So even though his model was wrong, the outcome was correct. And so maybe here you have the same thing, that what the tongue stimulation and auditory stimulation might have a benefit, but the explanation is wrong, just like in Thales of Mileto's story. And this is important to understand that, well, we should not always blindly believe whatever study is, but think about it logically. And if the results don't keep up with the story that's told, well, then we might have to, well, maybe the story is wrong. The results are the results. You can't change the data, but you can change the interpretation.
Wouldn't this mean that the, I guess, Lenire device, which actually is considered a habituation device, and this is one of the questions we have later on, so I think I shouldn't focus too much on this, would have a more profound effect on the emotional centers of the brain rather than, for example, inducing long-term synaptic depression?
Yes, but you have to see that long-term synaptic depression is at a synaptic level. What we look at a brain level, so a synapse is like if you go into a big soccer stadium or a football stadium, it's one connection, one person saying something to the else. That doesn't change the entire stadium. You need to have lots of synapses, I mean, in a stadium before whatever is being said, like people starting to sing a song together. You need a certain critical threshold of people, of synapses, in other words, before it has an impact on perception. And this is where, of course, it's always very dangerous to start at a synaptic level and to translate that, because these synapses, I mean, if there is two people who start singing as loud as they can, but there is still some noise in the state, nobody will hear them. So the danger of taking a synapse and bringing it to a phenomenological level, to a level where you look at clinical signs, is dangerous for that point of view. It doesn't mean it shouldn't be done, but you should have a huge effect also at a network level, macroscopic network level. And this is where I think both forms of bimodal stimulation should indeed look at what is changing on EEGs or on fMRIs, on an imaging study at a more global level that is easier to explain individual, that easier to explain the phenomena, the clinical picture than synapses.
Okay. All right. Well, I guess let's go ahead and move on.
Yep. So we've seen all of this. So then the question is, what is your understanding of spiked timing dependent plasticity and its role in neuroplasticity? Why are inventors like Lenire, which don't align with STD principles, compared to those that do? And should more tinnitus research focus on modulating fusiform cell activity in the dorsal cochlear nucleus, where hypersynchrony is key? I think we
I think we pretty much
more
covered all of these questions already.
Yeah.
Okay, we can.
Yeah, which is good. I mean,
yeah. We could probably skip this one.
Yeah. So I just, that's the summary, but I think you already gave the summary very nicely. So I don't think we should go in detail. But the summary is that the timing from a Bayesian perspective, and I'm a Bayesian, I love this Bayesian approach. It means if the presynaptic neuron, so if the neuron that brings information gives a signal before the postsynaptic, so it goes from A to B, from pre to post, then the brain might infer that this is causal. There is one signal that sends and the other one where it derives. Whereas if you get a signal in the presynaptic before it and the postsynaptic before, then it's acausal. So basically, if I speak and somebody else listens, then my speaker has to come before the listening. And then the second version, somebody's listening, but the person has not spoken, then we say, well, whatever he does has nothing to do with, because he couldn't know yet what was coming in as information. So that's the basis of spike timing dependent plasticity. And the figure below with the LTP and LTD is what you've explained already. And basically it means the further you go away from, so the further those two action potentials, the two signals are apart, the less they become linked. And that's what you said. Yeah, the timing is critical and goes, and the closer they're timed, the stronger the influence. And this influence this is under influence of acetylcholine and dopamine that have a different effect. And this is important because if you try to combine somatosensory system and auditory system, then you have to try to understand the microanatomy and see how granule cells bring somatosensory or non-auditory input via the cartwheel cells to the fusiform cells, and then the fusiform cells integrate this non-auditory or somatosensor input with the somatosensory with the auditory input that comes from the auditory ganglion cells, and there, of course, they compute what you see in the figure on top, which one comes first in comparison to the other one, and then how big is the time difference, and depending on that, then they will create either a long-term potentiation, meaning the nerve cell will become more sensitive to this, or long-term depression, where the nerve cell will become less. And this potentiation is then critically dependent on your glutamate, on your NMDA receptors, etc. So this mechanism is very well understood. And conceptually, it makes a lot of sense from a Bayesian perspective. It basically means if A comes before B, then A is the cause. And if there is an integration that goes wrong in timing, by creating long-term potentiation, which is then linked to the tinnitus, then we should do the opposite. We should disconnect the information that comes from the somatosensory system and the auditory system, and that's exactly what Susan Shore has done in developing a device like that. But the critical assumption is that what happens here at a very tiny level then becomes basically explodes into a kind of avalanche that will disrupt this at a much bigger level, where you also have to see, well, how can we take the salience away? Because even if it's a tiny signal that's very important, we will still perceive it. And this might be the reason why the results were not as good as I had hoped for. Maybe because she looks at it from an initial point of view, but she might have to integrate in her stimulator something that tries to take away the salience, or tries to also influence the emotional component, etc. But it's a very good start, and it starts, and this is very important, from physiological levels that we understand.
Okay, what an excellent explanation. Let's move on.
So another reason, but that might actually bring us too far, is that we can conceive of the brain as a complex adaptive system. The only point that I want to make here is, why can we still believe in Susan Shore's device, which looks at synapses in comparison to whole brain networks like the auditory cortex and the emotional circuits? That's because every complex adaptive system, whether it is a brain or the economy or the internet, has the same characteristics. First, they're complex. Second, they're adaptive. That's the definition of the system, complex adaptive system. They're self-organized, meaning there's not one single part of the brain that tells the other parts what to do. But the important thing here is self-similarity. And self-similarity means that just like in the fern that you see, that part of the fern has the same shape and might have the same function as the whole fern. So that means that what we understand from a synaptic level, like in Susan Shore's, might in some way be translatable to the entire brain. And so this is why you can only say that Susan Shore's work makes sense at a synaptic level if you believe in a complex adaptive system where you have self-similarity, that synaptic changes are somewhat similar or akin or linked to changes at another scale, at the level of the auditory cortex and emotional cortex, etc. And importantly, a complex adaptive system means that if you change the network, then you change the percept. So you can go from a tinnitus network. If you make the tinnitus network fall apart, then the tinnitus will be gone. So the criticism that you cannot use synaptic data at a glial level can be actually negated by accepting the brain as like an engineer, as a complex adaptive system. So here you have, for example, let's say this is a functional module for hearing. Then if this is a tinnitus module, that's a network that creates tinnitus, then that's easy to detect. You can just use an MRI. However, if you look at the transformer game with the same building blocks, so with the same dots that are put together in a different way, meaning their connections are different, then you can have the same network that's put together in a different way. So the same building blocks that are put together in a different way give two different emergent properties, meaning that your functional network and your tinnitus network might actually be extremely similar, except that the connections are different. So the information flow goes from one part to the other in a different way. And so this also explains why, if you change the connections by changing long-term potentiation or long-term depression, you change the connections between different parts of the somatosensory tinnitus network that you can relieve the symptom. So then there is a question about habituation devices and psychological interventions. The question is, why are expensive habituation devices promoted when their primary benefit seems to be psychological? Well, it's not only the primary benefit. We know that for Lenire, there is no change in loudness percept. So it's all about the suffering. It's all about how the impact. And to be realistic, I think these expensive habituation devices are probably on the market because some people just prefer to look at their problem, like I mentioned, like a complex adaptive system, like an engineer would look at it, rather than to look at it from a psychological point of view. And so it probably is geared towards people with this more mechanistic kind of mind, rather than people saying, well, I have to go to the psychologist to do cognitive behavioral therapy, because there's at least meta-analyses that show that it works good. It has a 10-point reduction, which is the same as the Lenire's device and Susan Shore's device. So it gives the same result, but via a different pathway. And some people just don't like to go to the psychologist because of stigma or whatever, and they prefer some mechanical thing to do the work for them. So I think that's why these habituation devices exist. Furthermore, if you live in the United States, going to a psychologist will very quickly amount to the same amount of money as a device like that. So I would assume that it's more a matter of what the personality is of the person with tinnitus, rather than anything else. And then the second. Sorry.
Oh, Hazel.
I also wanted to say something. No, because I think for me, the problem with Lenire, yes, now when you listen to Hubert Lim, for instance, making a presentation, he will literally say it's a habituation device. They didn't say that very clearly in the beginning, and their marketing materials also didn't make that clear. And that's where I do agree with the tone of this question, that, you know, considering the price plus how it was marketed, I do think a lot of people bought into something that, you know, really they might have really regretted later.
Well, I have kind of an opinion about this, and I think Dirk made an excellent comparison. There's just a certain subtype of people who do not positively, positively react to the fact that you have to put in a lot of work. For example, like in cognitive behavioral therapy, they would rather spend the money on something that, generally speaking, does not actually decrease tinnitus objectively, but, kind of overall improves their mood. And even if it's a placebo effect, like if a person wants to spend $3,000 to $4,000 on something that will make them feel better, then why not? All power to them. The real issue arises is when these things are peddled towards catastrophic sufferers who truly needs different interventions. That's the biggest issue I have with it. So again, if somebody wants to spend a lot of money on something that they think will help them, and eventually it will help their emotional state, then yeah, they might as well just go ahead and do it.
Yeah, and so then the next question is this not a form of exploitation, given that CBT could achieve similar results at a lower cost? Yes, of course. That's also that is a matter of how you see the results, and also probably, as we discussed earlier on your bias. Like we don't have a lot of patients that come to see us that are still are still candidates for it, because we've tried every kind of neuromodulation, and if it doesn't work, then the chances of it not working are fairly high, even though we have no hard data on it. With the Lenire device, we do have data that compare TMS and TDCS, etc., and C2 stimulation, to how much one predicts the other, and it can go up to 60% if you respond to one, that you will respond to another kind of stimulation, but not the other way around. So there is a correlation there, but we don't know. We haven't looked at it for Lenire specifically. And given that CBT could achieve similar results at a lower cost, yes, that probably depends on the country you live in. And second of all, I think there is probably an underrepresentation of CBT, maybe not in all countries, but if you look at it from a global scale, then there is probably not enough CBT specialists to tailor to the need of even tinnitus patients. So I think, and that's also why even if it's an expensive habituation device, why there is a market for it from a commercial point of view, because there is just not enough CBT specialists, and in the US prices are probably fairly high as well. And so the reference for this is that over 80% of the clinical benefit of the latest Lenire trial, which is the one I mentioned earlier on, where for the FDA, they had to do auditory stimulation and then look at the outcome, and then afterwards add the tongue stimulation and see whether there was a substantial benefit by combining them, irrespective of the mechanism of action. The statistical acrobatics in this study are deplorable. And why are not more tinnitus researchers calling this out? Well, the answer is very simple. The answer is that these statistics and the design was decided by the FDA. Basically, they said, well, you have to do this and this. And of course, this came as a little bit of difficulty for a company that says, well, gee, we don't know if we combine it, it's really going to be better than just auditory stimulate. Well, because the statistics was enforced by the FDA, basically. So that's why people are not calling this out, because, well, if the guy who's going to pay for it, well, or allow it, decides what the criteria are, then we can say, well, we don't agree with it. Just like the FDA asked Susan Short to do minimum masking levels. If they would have asked me, I would say this makes no sense whatsoever, because it doesn't correlate with the subjective loudness percept. But if that's what the FDA wants, that's you might say, well, this doesn't make any sense, but they want to see those so-called more objective numbers. But from a clinician, it doesn't make any sense. So that's the reason why no more researchers are saying, well, we don't like this study, because, well, it was decided by the FDA.
Okay. Yeah, that makes a lot of sense. All right, let's move on, I guess.
So this was the study that we were discussing. So first phase, only sound, then, and you see in gray the activity of the sound, and then the green was the extra benefit or no benefit. Like here, this patient had no benefit from the extra. This patient had also no benefit. Here we've got a couple of patients who got worse. But of course, that's a group level analysis, and on average, you see the green that there is extra benefit, but that does not explain the mechanism of action. Mechanism of action was just a hypothesis, and I kindly disagree with the mechanism of action. I think the mechanism of action is different, as I mentioned before. So then the tinnitus and hyperacusis relationship. Why isn't more research dedicated to loudness, hyperacusis, given its prevalence in severe tinnitus cases? And why are neurologists only now being told to stop using the term phonophobia for hyperacusis? And why hasn't this been corrected sooner? Why don't tinnitus researchers draw more from neurological studies or collaborate with neurologists on phonophobia, migraine, and TBI patients to better understand hyperacusis? So let's first address these questions. So why isn't more research dedicated to loudness hyperacusis? Well, scientists don't like something that doesn't have a definition. And if you look at, for example, the prevalence of hyperacusis in David Bagley's study, is looking at the prevalence of hyperacusis, it went from 7 to 70%. Basically, it means nothing. Then Smith looked at it and she said it's about 15%, which is very similar to the prevalence of tinnitus. And then in severe tinnitus, Siderot mentioned 80% have it. So basically, scientists in general, or research in general, like something that's well defined, that they know, okay, this is it. But because there is so many different definitions, then the question is which one you take. Well, whichever one you take, somebody else will say, no, you shouldn't have used the hyperacusis questionnaires. You should have done the sensitivity testing, etc. And so people kind of shade away from these before there is a kind of a consensus. Okay, this is the way we will study it, and these are the outcome measures. Then that will already boost. And this is a task for the TRI. We started in the simple way, trying to find a consensus, even though there's no general consensus, but some more or less consensus about the definition for tinnitus. And we have to do the same thing for hyperacusis, misophonia, phonophobia. And what's the difference between all of these? In simple words, phonophobia is, well, if you translate it literally from Greek, it means fear of sound. Hyperacusis is hypersensitivity for sound, and misophonia is a dislike of sound. So, of course, these are kind of related in the sense that if you're very hypersensitive to sound, that might lead to fear of sound. If you say, well, whenever I go to a restaurant, I really get my hypersensitivity doesn't allow me to join it. Then if this keeps on being confirmed, then your brain will become fearful or might become fearful of it, and that's why probably people used it intermittently, even though they actually mean something different. So they're related, but they're not the same.
I think the main reason of why hyperacusis, specifically loudness hyperacusis, is so undermined in research is that loudness hyperacusis does have at least from the data that I've collected over the years of studying the community, it does have a very good track record of improving with time. I would say that it's actually very significant. I'm just throwing numbers out there, but I would say it's maybe over 80%, maybe even higher. So maybe people who, maybe the researchers don't think that this is enough of an issue to research, since it has such a good recovery rate. Considering that, stuff like tinnitus training therapy or, sound enrichment is also seen to bring benefit to it, which I kind of disagree with, but there has been some data that shows that a very careful reintroduction of sounds, natural sounds, does help hyperacusis, loudness hyperacusis in the long run. So yeah, that's my take on that.
No, and you're perfectly right. Of course, also from a research point of view, if I say, well, I'm going to research whether treatment A benefits hyperacusis, then the first reviewer will say, yeah, very nice study, very well designed, but could this not just be the natural evolution that they spontaneously evolve? Of course, it's very different if you do a very short acting study, then that's very different. But if you do a longer term evaluative study that takes a couple of months, then of course, and it will be very hard because then you have to have a control group that runs in parallel and you follow it for months to see that there is a difference. So it makes studying it hard, and researchers are or also trying in general to take the path of least resistance, and not go for things that they spend lots of time and energy and then might be killed by the first reviewer who says, well, sorry, I consider this a natural evolution. And so there's questions about Noxacusis. One is, why is there so little research into Noxacusis, which is painful hyperacusis, so where an external sound is not only perceived louder than it should be, but also as painful. A condition that clearly involves the trigeminal system and causes immense suffering. There is probably a couple of reasons why there's little research into Noxacusis, because it's not that extremely common. So if you want to research it, you need to either have a large clinic from where you can see a fair amount of people with tinnitus and associated Noxacusis, or people where the Noxacusis is the predominant symptom and the tinnitus is there, but it's not as bothersome as the Noxacusis, or you have to do research in a highly populated area where you can also find many people. That's one of the reasons. The other is that it is not as easy to treat as tinnitus, in the sense that in tinnitus we've got a great approach where, like in the tinnitus questionnaire, you have 4 grades or 4 levels of how much it bothers or how much stress it causes, whereas Noxacusis is not very well known, rare, and not very well defined. So it's just the fact that people don't really know it very well, that it's very little researched, I think. I don't know if you have any other reasons why.
Honestly, I think I can completely agree with you. Noxacusis patients are very, very not prevalent in the community. Well, no, there's quite a bit of them, of course, on the forums, et cetera, but as we all know, forums are not a good source of data, specifically on like who's suffering from what, because people who have more severe conditions tend to stick around longer. So yeah, I think I can completely agree with you there. That makes sense. Yeah.
And then the second question is, why are patients with Noxacusis routinely casted by the clinical community as having psychological issues rather than receiving proper medical investigation? Well, one of the reasons is, of course, that it is that these patients severely suffer, and therefore for clinicians sometimes come across as very demanding, in that they need to be treated very urgently, very quickly, very aggressively, and that clinicians tend to shy away from it, saying like, oh, this is too demanding, I'll just go to the more easily treatable patients. And also, receiving proper medical investigation is not easy, because there is no proper standardized approach to treating Noxacusis. So even the terminology is not generally accepted. So if it's not very well defined, people don't know how to investigate this in the proper way. How do you correlate the loudness of a certain sound to how much pain it causes? I mean, this is doable to standardize the approach, but it just hasn't been done yet. And therefore, in the clinical world, if we as clinicians don't understand or know something very well, then it's probably easier for us to put it into the psychological or psychiatric realm and saying then psychologists and psychiatrists have to take care of it rather than us. So it's probably a lack of understanding of a somewhat less common pathology.
Yeah, I really wish that, well, I'm not addressing the entire community of clinicians, but I really wish that they would utter the phrase, I don't know, or they would have the ability to utter the phrase, I don't know, instead of offering I don't want to say gaslighting because maybe from the perspective of a clinician, it doesn't seem like gaslighting because they're truly, I guess, trying to help out and, automatically directing their patients towards psychological care would make sense for them in that moment. But I really wish they would admit to not knowing something instead of making stuff up and saying that you're crazy or something like that. Okay, let's move on.
So Dysrhythmia is even a bigger problem. If you look at auditory distortions and you just type in PubMed, which is the kind of the standard NIH-sponsored repository of scientific articles, then there's only 17 papers on auditory distortions. So virtually nothing is known about its mechanisms, how they develop. And in general approach, if we see patients with tinnitus or hyperacusis with auditory distortions, we treat the tinnitus, and often the auditory distortions improve when the tinnitus improves or when the hyperacusis improves. So you can, well, the prevailing idea is that it's a problem of the wave inside the cochlea, and that therefore activates different fibers that brings information from the cochlea to the brainstem and from the brainstem to the brain, and that because the wave is not properly formed, that results in normal transmission of the sound waves via the different nerves, and that causes the auditory distortion. Of course, this is one model, but you could also imagine that the information is transmitted correctly, but is distorted somewhere along the trajectory to the auditory cortex, and from there to the to Wernicke's cortex for understanding and interpretation. So there is a lot of possibilities where it exists. And as clinicians, often you hear, okay, I hear distorted sounds, and then if you treat the tinnitus and the hyperacusis, these distortions seems to improve as well. And so because there is very few patients, at least in our clinic, that come with only auditory distortions. They might be more common in the ENT or the audiology world, but in our clinic, which is predominantly brain focused, of course, well, we don't see those.
Mm-hmm. Yeah, I absolutely agree with you with the lack of research. And over the course of the past few years, I have attempted to do this, to research Dysrhythmia, and there has been actually one individual in the community who has studied this even more extensively than I have for over half a decade, and they told me that there's over quote unquote 300 potential causes. Yes, and that's an absolutely phenomenal amount of causes. And yeah, you did mention that Dysrhythmia or distortions do tend to improve with tinnitus and hyperacusis improvement, which is visible actually, obviously. And I think my personal theory is I think that there are some kind of, I guess, well, I don't want to go back to the fusiform model, but it does make sense to take a look at it. But with the extreme hyperactivity or the heightened hyperactivity, the firing of action potentials of the fusiform cells, et cetera, and the dorsal cochlear nucleus, potentially the pathways of fusiform cells that are responsible for different frequencies can actually become engaged when they're not supposed to be, just because of this extreme heightened hyperactivity, and there can be, I guess, some crossfire or something like that. I mean, I guess it would make sense to see it this way when some people have used the Susan Shore device and they have had their Dysrhythmia or distortions improve, I guess, to a significant degree. So yeah, I agree. There's just lots and lots of potential causes. Like you mentioned, the auditory nerve fibers, that's absolutely possible as well. And it's just too much and too little people actually experiencing it. Okay, I guess we can move on.
Yeah. So then we got into my preferred domain of thalamocortical dysrhythmia. So thalamocortical dysrhythmia is implicated in tinnitus, migraine, visual snow, as well as other disorders like Parkinson's disease, depression, slow wave epilepsy, schizophrenia, etc. Now, the question why tinnitus or thalamocortical dysrhythmia and migraine is not further investigated is probably a better question for John Lilian Hamid, who's a specialist in migraine and tinnitus. So I think it would be nice to have a similar question round with him on everything that's migraine related. Now, the second question is, how could understanding of thalamocortical dysrhythmia improve our ability to manage the temporal fluctuations seen in tinnitus? And that's a very good question. So thalamocortical dysrhythmia, basically what it does is normally your resting state activity runs in alpha speed. Alpha means that between 8 and 12 hertz. So everybody has an individual alpha frequency, which is like when you start your car and it's just the revolving activity of your car before you put it in first gear. Now this thalamocortical dysrhythmia then is caused by a lack of input, whether it's somatosensory input, auditory input, it doesn't matter. If there is less input arriving in the thalamus, it says, well, I don't have to process as much, so I will slow down from alpha to theta, which is a speed between 4 and 7 hertz, and it will just transmit that or relay that information to the cortex, and you'll get a small cortical blob of 4 to 7 hertz activity, which is similar to what you could call a sleep frequency. So when you're asleep, your alpha activity slows down to theta, and when you go to deep sleep, it slows down to alpha with delta, which is in between 1 and 4 hertz. So basically, it means that you've got a little blob on your cortex in the auditory system that fires at 4 to 7 hertz. Basically, it's asleep. But because it slows down, it suppresses the adjacent area less, and you get a hyperactivity zone, a halo, around the small, slower spot. And that's what we call thalamocortical dysrhythmia. This idea was developed by Rodolfo Llinas in 1999, published. And basically what he said is the theta represent the deafferentation, so the hearing loss in the auditory system, the sensory loss in the somatosensory system, the visual loss in the visual system, and then the surrounding high frequency gamma activity represents the hyperactivity. So the tinnitus in the auditory system, the pain in the visual system, etc. And so that's the essence of thalamocortical dysrhythmia. In the visual system, it's called visual snow syndrome. And of course, again, should we focus more on visual snow? Yes, if there is research money, it would be certainly interesting, but there is only 170 papers on the visual snow syndrome, so there is not a lot we can learn from the visual snow syndrome, as there is even a lot less papers on tinnitus. But probably visual snow can learn from tinnitus, as tinnitus can learn from pain, because the more publications there are from similar syndromes, the more we can learn. And then the question whether CGRP, mediated neuroinflammation, or TCD, can explain the overlap between these conditions. Well, it is certainly involved, but I'm sure that Hamid would be the key person to discuss the influence of CGRP, mediated neuroinflammation, which is very better studied in migraine than it is in tinnitus and in visual snow. So the pain literature, which is a lot more advanced, remember there is about more than 1 million papers published on pain, and a lot less in tinnitus that we can learn from a lot from pain. That does not always mean that we tried a lot of drugs that seem to work in pain and tinnitus, and most do not work in tinnitus or do not benefit tinnitus patients. So it's not because CGRP treatments like antibodies, etc., seem to benefit patients with migraine that they might also benefit tinnitus, unfortunately. So neuromodulation therapies for tinnitus. First question is, why has your team stopped publishing on TMS for tinnitus? And this is a very good question because we initially we did most of our studies on with TMS, and the idea was very simple. When we started with TMS, there had been a publication on a PET scan that showed that in tinnitus patients there was increased activity in the auditory cortex associated with tinnitus. So we said, well, if we can suppress this hyperactivity, we should be able to treat it. So then, and in keeping, and at the same time, Rodolfo Linas published this Thalamocortical Dysrhythmia treatment theory. So we said, well, if we treat this gamma activity, we should be able to treat the tinnitus. And so that's when we started using TMS. And we very quickly realized that even though it seemed to have some benefit in some tinnitus patients, it was certainly not the holy grail, which again then stimulated people to start looking for new theories. And that's when a couple of years later, Winnie Schlee came up with the idea that actually tinnitus was an emergent property of a network phenomenon, which is in keeping with the complex adaptive system that we discussed earlier on. And so this problem with TMS is, first of all, that the clinical benefit was not very dramatic, and second of all, is that it can only target one area. And so once you conceptually change from the idea that tinnitus is related to one area in the brain and the auditory cortex that can explain everything, and you try to suppress it by transcranial magnetic stimulation, by implanting electrodes, and you see that you cannot cure everybody, then you have to change either the approach or the theoretical model. And so when Winnie Schlee then suggested, well, actually it's not a single area, it's a network model, then we thought, well, how can we change a network by following a paper that was published by Albert and Barabasi that said if you want to change a network, you want to disrupt it, you have to target multiple hubs, meaning the most densely connected parts of that network, which means a single target is not good enough. So initially we tried it with 2 TMS machines, which was not very easy. And then electrical stimulation became again more in vogue. And so we kind of switched to electrical stimulation because there it is a lot more easy to target multiple areas with the new TDCS machines that can have up to 32 independently controlled channels, which means you can fractionate or you can divide the current through different electrodes, and depending on the strength of each electrode, you can send the current to one area to activate it, to another area to inactivate it, to a third area to do nothing. So you can really rebalance different networks in the brain. And this is where these new devices that are highly sophisticated and depend on a lot of computational modeling actually allow us to better understand the physiology and modulate the current understanding of the different networks that are involved in the brain. So that's why we kind of stopped doing TMS research and are more focusing on electrical stimulation right now.
Okay,
And then the last
good explanation. Got no questions. Let's move on.
So then the question, what about enhancing the descending noise cancelling pathway, for example, through the vagus nerve stimulation or TDCS, provide meaningful relief for chronic tinnitus? I do believe so. We've published papers where we hypothesize based on the auditory system, somewhat the sensory system and pain, that actually whether you have tinnitus or not is not always related to more input, tinnitus input, but also a lack of suppression. And to be honest, this was not our idea. It was Rauschecker, his idea, who came up with this noise cancelling proposition. And we fairly quickly afterwards thought about it and said, well, maybe it's not all input and it's not all noise cancelling or all noise cancelling, as Rauschecker put it, but it's a balance between the two. And if it's a balance, that creates some very intriguing ideas that you can have a reduced input, but also an even more reduced cancelling, and then you still have tinnitus. So you can have more input and suppression of noise cancelling. That clearly gives more input, so more hyperactivity, so more tinnitus. But you could also have decreased input as long as the descending pathway is even more suppressed, then that will still give a relative balance in favor of more input than suppression. And so this allows to explain way more imaging results where sometimes the auditory cortex is hyperactive, sometimes it's hypoactive, and people really got confused. But this balancing idea does make it more easy to explain. But fundamentally, what it means is if you compress the brake, which is the descending noise cancelling pathway, you're always good in suppressing input. And therefore, there has not been one single formal study, even though we routinely use it in clinical practice, that investigates this in more detail. You can reach to the pre-general anterior cingulate cortex, which is the starting point of the descending noise cancelling pathway via the dorsolateral prefrontal cortex, which is the traditional target for TMS and the traditional target for TDCS. We do know you can reach it and we use it, especially in those people who have no hearing loss, because we've seen in one of our studies that there might be at least two types of tinnitus, one with clear hearing loss, which is more auditory cortex and parahippocampus dependent, and one without clear hearing loss that seems to be more depending on a lack of noise cancelling. So yes, you can enhance descending noise cancelling via bifrontal stimulation with TDCS, via anodal stimulation prefrontally, and via other mechanisms, and it is used in neuromodulation clinics.
Okay.
So just to show that the results, if you look at TMS analysis, it's about the same as what you have for all the other treatments. You have, there is evidence, meta-analytic evidence that shows that you get a benefit on the THI, which is 7 to 8 points, which is similar to Susan Shore's results, which is basically the same as the MC idea. So it's only a minimal clinical benefit at the group level, of course. but there is no real loudness reduction. So it does seem to improve the suffering, but not the loudness. When you look at electrical stimulation, there is only studies on the traditional approach, which is the bifrontal stimulation, and it improves loudness. But look at the benefit; it's minus 0.35 points, which is less than the minimal clinically. So statistically, it's significant, but clinically, it's an irrelevant improvement of loudness. I mean, if your loudness drops from 9 to 8.65, I wouldn't really call that a clinical benefit. And the distress also improves, but that's different. Other studies do show, it improves the stress, but not the loudness. So ultimately, what is really interesting here is that if you look at a network meta-analysis, which looks at all the outcomes and then kind of looks which one is better, then what you see is that if you target both the auditory cortex and the frontal cortex, that you get a better benefit, which is in keeping with this network approach, that if you target multiple nodes simultaneously, then the network falls apart better. So yes, neuromodulation benefits. The benefit is about the same as for cognitive behavioral therapy, the same as for bimodal stimulation, the same, but not as good as, at least according to the studies, as the Lenire device, because that is a little bit better, according to the studies, but and about the same as whatever has been published, really. So it's not better, it's not worse, it gives the same amount of benefit. Now, remember that we talked about thalamocortical dysrhythmia as being one mechanistic explanation for both tinnitus and pain. And then a question, of course, arises, can we learn from chronic pain what we learned for neuromodulation chronic pain or pharmacotherapy to be adapted as similar pathways in tinnitus? And this has been the mainstay of not only our research, but of many tinnitus groups that also look at pain. And the first studies were done at the end of the 20th century by Tondorf in Germany, and then by Agamöller, who was my PhD promoter, and who said, pain and tinnitus is exactly the same, except, as I mentioned earlier on, that there is a specific pain pathway, but there is no specific tinnitus pathway. So acute pain and acute tinnitus are different, but the chronic forms might be fairly similar. And so, yes, the neuromodulation approaches are the same. The pharmacotherapy approaches are very, very similar. But there is more drugs that work for pain because there is a pain-specific pathway than they work on tinnitus. So tinnitus tends to be more difficult to treat than pain. And if we see patients who have both tinnitus and pain, actually, in general, the pain is easier to treat than the tinnitus. So even though it seems to be simple, I mean, it's just a sound that's present without a corresponding external sound source. So conceptually, it's a very simple percept. It's way more easy than hallucinations but in reality, it does cause humility in researchers, because I remember when I started, I thought, well, it seems to be so simple that within 5 years we should have found a solution, and 20 years later, we're getting closer, but there is still no universal solution for tinnitus. Another question is, why isn't there more research into circadian rhythm disorder and tinnitus? The reason is because it's difficult to research. Circadian rhythms require either genetic research, which becomes easier and cheaper to do, but also knowing what to look for. And I think we're just in the first stage now with researchers like in France, Robin Guillard, who got a PhD in looking at sleep and tinnitus. And of course, sleep is not the same as the circadian rhythm, but it's one of the circadian rhythms is a day-night cycle, and sleep, of course, is linked to that. And this is just an emerging field right now, where we see there is a correlation between the sleep architecture, so the rhythm of the sleep, basically, because sleep is not just universal state, it has up and down states that, depending on how much deep sleep and REM sleep you have, actually that determines how loud and how bothersome the tinnitus is on the next day. So it's clearly important. Also, if you know that 50% of all our genes are triggered or expressed in following a circadian rhythm, it is very important, especially for people who have associated immune problems, which could be allergies, but which could also be more in the setting of associated depression and anxiety, as we've seen that these are neuroinflammatory pathologies. So the link between tinnitus as a neuroinflammatory disorder and a neurological expression are probably to be found in the circadian rhythm. And the reason is relatively simple. Our brain consumes about 25% of the energy that we consume. Our immune system, about the same. Our gastrointestinal system, about the same. And our heart and lungs, also about the same. And on top of that, because then we're already at 100%, we've got extra energy to be used for muscles and movement, etc. Now, so what happens when we don't sleep enough? Then the problem is our immune system, because our immune system is predominantly active at night. And the reason is that during the day, we use predominantly our brain, so we don't have as much energy for the immune system. And at night, our brain consumes less energy because it's asleep, and then there's more energy to be spent on the immune system. So if we have tinnitus, and more than 60% of people with tinnitus develop sleep disorders, that will result in an immune problem, and the immune problem can then cause a neuroinflammation, which causes the tinnitus to become permanent. So it is clearly a very important topic, but it needs to be studied from a genetic point of view, as well as from a sleep research point of view. And that's not always easy, because you have to be able to study sleep. Up to now, that was done by polysomnography, with the higher accuracy now of sleep following devices like the Oura Ring and Whoop and Apple Watch, that have, depending on which kind of tool, wearable you use, you can have a fairly accurate way of monitoring sleep and circadian rhythms. Actually, this research will start to be developed in the very near future, because now it becomes relatively easy to do in comparison to the very expensive and difficult polysomnographic research that needed to be done up to now. So why has there only been one study showing a correlation between tinnitus severity and hair cortisol levels? And shouldn't stress and its biological markers be more thoroughly explored? Yes, it should, but this is a huge specialty in itself, in the sense that acute stress causes cortisol levels to increase. chronic stress is more difficult, and the response to cortisol in acute and chronic stages is relatively different. So if cortisol would have been the perfect marker of chronic stress, which is more commonly associated with tinnitus than one pure acute stress level, then it would be studied more commonly, because it's relatively easy to do. You can take saliva or blood, but saliva is probably easier. But it's not a very good marker of chronic stress, and therefore it's not being studied that commonly. So it's probably because it's not the optimal stress marker. There is actually not yet one optimal stress marker. For example, people often say, well, we should use HRV. Well, we monitor HRV in every single patient, but HRV represents predominantly parasympathetic, so rest, digest, and restore activity. And it is commonly assumed that the parasympathetic and the sympathetic system are anti-correlated. So if this fight and flight response or stress response from the sympathetic system is active, the parasympathetic system is suppressed. This is a very old, typical Western approach of thinking. One is up, the other is down. You either win or you lose. But in reality, in the body, it's more like an Eastern philosophy where it's a yin-yang idea, where it's a little bit of both. And on top of that, we don't even know if the balance could be broken, if the sympathetic could be down and the parasympathetic could be down because the balance is broken, or both could be up. So we clearly don't understand something as fundamental as the stress system well enough, because of we don't have any very good biological markers. But with big data, that would become easier to look at. So pupillometry might be one. Skin conductance is too slow, but there is now a new measure since a couple of years that is instantaneous. So it can look at acute stress levels. It's just a computation of a heart response. So it's easy to do, but it's not yet commercially easily available. So once we have got good biological markers, a lot of research will be done. But cortisol is not one of them. Then another question is, how might the parallels between ADHD and tinnitus, two conditions that affect the Salience network, inspire development of new research and treatments? Well, we've just published a paper that was accepted today, actually, on not only ADHD and tinnitus, but on thalamocortical dysrhythmia. So where we looked at tinnitus, what do tinnitus, pain, Parkinson, depression as thalamocortical dysrhythmias have in common with ADHD, alcohol addiction, food addiction, and which are all thalamocortical dysrhythmias, and OCD. So these are the reward deficiency syndromes. And what we found is actually that there is a common activity in the dorsal-anterior cingulate cortex, and there is an opposite activity between the two groups at the pre-general anterior cingulate cortex. So one is a lack of suppression, and the other one is too much suppression, basically. And the idea is here that what they have in common can probably be brought back, and now this is hypothetical, at the very at the receptor level, where the reward deficiency syndromes are based on a genetic common abnormality that is expressed in different ways. One way is ADHD, another is OCD, another one is addiction. But they have common genetic polymorphisms, so genetic variations, and that's how it was defined. And initially it was only on the dopamine D2 receptor, but then it became also the serotonin 1A receptor, which is intriguing because that's the one that can be improved by psychedelics, and also some other neurotransmitter receptors. Now, when we looked at it, we saw that thalamocortical dysrhythmias, which is characterized electrophysiologically. So the one is a genetic group of pathologies, the other is an electrophysiological group of pathologies. But when you look at ADHD, actually it also has thalamocortical dysrhythmia electrophysiologically. So it has theta gamma abnormalities. And so basically, we think that they're actually two sides of the same coin, where in one, the genetics is more stressed than the other, the electrophysiology, but ultimately they're the same of the same coin, which is very nice because by the fact that we could show that tinnitus, pain, Parkinson, depression, ADHD, addiction, and OCD have some common pathophysiology, that means that we should not only borrow treatments from pain, but we can borrow tinnitus treatments from ADHD, we can borrow it from OCD, we can borrow it from addiction. So that might open up more treatment possibilities for tinnitus in the future. And of course, as correctly mentioned, the two conditions affect the salience network, and the salience network is what we showed that they have all in common, which basically means they're suffering. And so we think that what we see as a common effect is the fact that they suffer, that they all suffer, but from something different. And so that if we target the suffering, which is not the optimal, but it's second good, that we should be able to treat suffering in all these pathologies in a similar way. And of course, you can't suffer from something that's not salient. If you don't care about something, something doesn't have any behavioral relevance, if you don't care you have tinnitus, then you won't suffer from it. If you don't care you have pain, you won't suffer from it. And intriguingly, this target that is common between all these pathologies is the target for cingulotomies, so for a small version of a frontal lobotomy that really removed the suffering from all these different pathologies. And people in the 1940s, when they did those lobotomies, had no clue what they were treating, but it seemed to work, and especially on the suffering. So in tinnitus, the patients would typically say that it's equally loud as it was, but it doesn't bother me that much anymore. And in ADHD, we know that part of the driver in ADHD is actually in the same area. So how do you interpret the similarities between tinnitus-related cognitive issues and ADHD? Are there clear distinctions to be made? No. For that, we might have to resort back to what we discussed earlier on, a triple network model. So remember that the salience network shifts people's attention to default and to default mode, which means internal cognition, and central executive network, which means external cognition. Now, this is pathological in both ADHD and tinnitus. And so basically the ADHD brain can't distinguish behaviorally relevant from non-behaviorally relevant. So it will resort to a better safe than sorry approach and say, well, we consider everything behaviorally relevant. So every stimulus that arrives is being focused on. And therefore, people with ADHD are constantly running from one stimulus to the other stimulus without being able to focus on something that's really behaviorally relevant. And in tinnitus, you have the opposite, where the brain hyperfocuses on the tinnitus, and therefore that takes away important attentional resources from competitive stimuli. And so this is where, remember, in ADHD and tinnitus, you've got the opposite functioning of the pregenual anterior cingulate, which is the descending noise cancelling pathway, which is not functioning very well. And in ADHD, there is also no stimulus suppression. So there is commonalities between the two, and therefore also there could be potentially similar treatments.
Okay, I think that is an excellent description. Let's move on.
Yep. So when we look at the first question, what can we learn from treatments for chronic pain? Then if you look, it's a little bit too small, but if you look at all the medication that has been tested for tinnitus, and this long list on the right side, they're all medications that are either used, or predominantly used in pain. So most of them have been actually copied from pain to tinnitus. But unfortunately, there is not one single drug that has acquired FDA or CE approval for the treatment of tinnitus. Whereas, of course, there is a lot more that are commercialized and accepted as treatments for pain. So the problem is that 10 years ago, a couple of studies were done, meta-analyses, that looked at the benefit of antiepileptics, antidepressants, ginkgo, and there was not one that generally improved tinnitus. And this is the weakness of meta-analysis, because you lump everybody together. And remember the earlier discussion we had on food, where there won't be any food, any nutritional approach that treats every tinnitus patient, because it depends on their allergies, it depends on their hypersensitivities, it depends on their microbiome, etc. And you can say the same from antiepileptics and antidepressants. Of course, if you take patients, let's say you take the first 200 patients that you see, and you do a study, and you happen to be in, let's say you happen to work in a department where you see only grade 1 tinnitus patients, and you give antidepressants, it won't work, because they're not affected psychologically by their tinnitus. Whereas if you go to a tinnitus clinic like ours, where we see a lot of grade 4 patients, then of course antidepressants might give a benefit to those. So again, as was mentioned by Anthony earlier on, that the bias is big, whether you want it or not. And when you lump people from different studies together at a meta-analytic level, then the chances that you find something get even smaller, especially if you don't know subgroups, or if you cannot stratify in a granular way the different pathologies. So currently there is no medication that's FDA or CD or CE approved for tinnitus. That doesn't mean nothing works. And as we mentioned before, nothing works in humans. Irrespective of that, a lot of medication seems to work in rats. For example, memantine, carbamazepine, baclofen, as I mentioned earlier on. These work in rats, but not in humans. I also mentioned on another question on the circadian rhythms that sleep is important. So here is one of these studies that was recently published that shows that there is a difference in N2 sleep, there is a difference in N3 sleep and in REM sleep. So if you have not a lot of deep sleep, which is N3 sleep, or REM sleep, which is what we traditionally call dream sleep, then a lack of deep sleep and REM sleep correlates with worsening of tinnitus, whereas more N2 sleep correlates with less tinnitus. And here you see the correlation coefficients, where if you have less REM sleep, then actually the tinnitus gets worse with REM sleep, and the loudness rating of the tinnitus intensity variation. So basically it means that yes, sleep is very important, and there might be a circadian rhythm that determines how bad the tinnitus is on the night afterwards. And then one of the last questions
a bit before we continue,
is,
There's been a paper that not a lot of people have been paying attention to, unfortunately, by Jingquang Tang, that has showed that there is a serotonergic regulation of excitability of principal cells in the dorsal cochlear nucleus. And it was discovered in this study that serotonergic fibers are actually concentrated in the dorsal cochlear nucleus. Unfortunately, it does remain unclear what is the function of this dense input, but this study actually discovered that 5-HT directly enhances the excitability of fusiform cells via the activation of 5-HT receptors. And I wanted to hear your opinion on this matter, whether or not this actually would, I guess you could say, demotivize people from using, using antidepressants or any SSRIs in the treatment of tinnitus, considering that, the previous things we discussed with DCN, hyperexcitability, hyperactivity, synchronization, and so forth.
Yes. So of course, as you know, the principal cause of the serotonergic pathways is in the dorsal raphe and the brainstem, but there is a pathway which we've discussed earlier on, the descending noise cancelling pathway. Is partially serotonergic, partially noradrenergic, partially dopaminergic, and partially opioidergic. So basically, nature has caused this pathway that has multiple influences to be able to modulate the activity of the sound pathways. And so to put it very simple, sound pathways are excited by glutamate and are inhibited by GABA. So you've got an accelerator, which is glutamate, you've got a brake, that's GABA. All the other neurotransmitters like serotonin, dopamine, etc., are what you could call modulators of this inhibition and excitation. Or to put it into metaphoric way, these are like shifting back gears. So if you want to slow down a car, or translate it, if you want to slow down hyperactivity in the brain, which we know is present in the cochlear nucleus in tinnitus patients, if you want to slow this down, then what we would intuitively think is, well, we have to press the brake, we have to increase GABA, we have to release the accelerator, which means we have to use glutamate blocking agents like perampanel or memantine, and we have to shift back gears, which is blocking dopamine, activating serotonin, and activating opioidergic mechanisms. So from a theoretical perspective, we can look at that and say, okay, we need a cocktail. We don't need one product, we need a cocktail. Now, the problem is that dopamine has 5 receptors, serotonin has 7 receptors, and it's the balance within the dopamine, especially D1 versus D5, or D1-like, which is D1 and D5 versus 2, 3, and 4. That's dopamine, that's very different. If you go to serotonin, it's 2, 3, and 4 that are most important of the 7 receptors for tinnitus. And they have opposite mechanisms. So you can't say that we have to increase serotonin or we have to decrease. No, it's a fine-tuned balance between activating specifically 2 and 3 and blocking 4 or the other way around. So, and this has been studied in pain, and so we've used this to use aripiprazole, which we haven't published yet, but it is based on pain therapy, and it has the perfect mixture of receptor profiles that fit with pain counseling, and we assume it might be similar in noise counseling. So yes, serotonin is important, but what's even more important is which receptors the molecules modulate. So it's not all or nothing. Otherwise, we would not have 7 receptors, of course. We have 7 receptors because they all do something differently. And it's a balance between the receptors that determines whether it is good or not.
Okay, guess let's move on then.
Yes, so and we continue with treatments. What are your thoughts on the potential of stem cell therapies, regenerative medicine, cochlear stimulation strategies for tinnitus? Aren't these treatments fundamentally trying to regulate dorsal-cochlear nucleus hyperactivity and hypersynchrony? Well, stem cell therapies have been hyped and discussed about a lot. The problem with the stem cell therapies is not obtaining stem cells. That's easy. The problem, however, is a stem cell or a pluripotent cell is a cell that can become what you want it to become, whether it's a fusiform cell, a cartwheel cell, it doesn't really matter, on the condition that you know the soup of neurotrophic factors, so growth factors, etc., that then turn the stem cell into a cartwheel cell or into a fusiform cell or into whatever cell you want. And we don't know that yet. So we don't understand the soup that we have to make of those neurotrophic factors to turn it into one specific cell. Furthermore, once we've turned it into a cell, we have to block it from becoming a tumor, meaning from growing and continue to grow until it becomes a tumor. So the stem cell therapy is a theoretically very good idea. Unfortunately, we don't know yet how to control its growth and its direction of growth and blocking its growth. So ideally, you have a healthy soup of molecules that can drive it into a certain area. But the problem is that in pathological situations, the healthy cells have died because of, for example, hyper excitability, as we've seen before, with the C fibers that can become hyper active and actually kill themselves in the neighborhood. And that means that there is a lot of other molecules there to clean up the dead cells, but that's not the same cells that you need in order to grow new cells. So the biggest problem is not the stem cells, it's the knowing which molecules have to be at which site at which time. And so that's where stem cell therapy has up to now unfortunately failed because of the complexity of the growth factors. Similar to regenerative medicine, it's the same problem. Cochlear stimulation for tinnitus is very good. It has the highest success rate of all treatments for tinnitus. But the problem, of course, is that for intracochlear stimulation, you need to be deaf, or if you use a short electrode, you can still have some hearing left. But of course, there's 15% of the population has tinnitus, and most tinnitus patients still have good hearing. And therefore, intracochlear stimulation is not an option. There is now cochlear stimulation being developed that doesn't have to use an electrode to go into the cochlea, but stimulates the cochlea from the outside. But again, here, and even though I'm a neurosurgeon, so as a surgeon, I love surgery. The problem is that this will only be for very severe cases where nothing else helps, because you can't operate 15% of the population with a surgical device for tinnitus. So it might work, and intracochlear stimulation has the best outcome results of everything that has been done. But only for those people who are unilaterally deaf or bilaterally deaf. Aren't these treatments fundamentally trying to regulate DCN hyperactivity and hypersynchrony? Yes, they are, but they're also simultaneously modulating auditory cortex activity, also cochlear. The whole auditory pathway is basically modulated by these therapies. So they're certainly not exclusive to the dorsocochlear nucleus, but anything that is being done at the cochlea passes through the cochlear nucleus and therefore modulates the hyperactivity and the hypersynchrony.
Okay, now before we move on to the question that is regarding potassium channel research, specifically KCNQ2/3, targeting those specific potassium channels, I wanted to add something to this question regarding the efficacy of this treatment. There have been numerous cases, again, purely based on the experiences of the community, ergo just more non-clinical data, of course, but it has shown some potassium channel openers, such as, for example, reticabine, which actually targets way more potassium channels than necessary, leading to unsatisfactory side effects, have actually shown to significantly suppress the tinnitus objectively. And the thing that I wanted to add to this question was whether or not only the role of potassium channel research and how it fits into the potential tinnitus therapies, but whether or not this will lead to long-lasting changes. Because based on my understanding of potassium channel openers, since I guess you could say they have less of an effect on the synaptic connections between the neurons, whether or not this can even lead, or using them on a maybe pretty often, I guess you could say, could potentially lead to long-term synaptic plasticity or long-term changes in the dorsocochlear nucleus. That's basically what I wanted to add to this question about the long-term effect.
Yeah. So so we will talk a little bit about the potassium channels. So if you look at the potassium channels, then as you mentioned, reticabine is a drug that's not available anymore, unfortunately, because it's the only one that really works on those channels because of having too many side effects. But it does have a unique profile. So any nervous system, because this is now looking at all the anti-epilepsy drugs and how they work. Like some work on the sodium channel, some on the calcium channel, some on GABA, some on the glutamate. So basically, reticabine works on the potassium channel, but is not available anymore. So you could imagine that modifying the potassium channels influences the firing pattern, and especially the bursting. Well, not only the bursting, but also the bursting that is being linked, at least in animal studies, to tinnitus. Now, there might be another mechanism which I think is even more important, and this is what's called a GABA switch. So GABA, I just mentioned, is actually a inhibitory. It's the brake of our nervous system. However, when you're born, GABA is excitatory. So initially, when you're born, GABA is excitatory, and what it does is it allows chloride to exit the cell, and therefore the cell becomes less negative on the inside, more positive, and that creates hyperexcitability. But after a certain age, which is related, of course, to neuronal maturation, there is a switch and GABA becomes inhibitory. And this is linked to those KCC2 channels, where basically GABA does the opposite. So instead of chloride going outside the cell, goes inside the cell, and then it becomes inhibitory because it becomes more difficult to polarize. Now, hypothetically, and this has been shown in the vestibular system, and now Arnaud Norena in France is looking at that as well for the auditory system. When you have a sudden hearing loss or sudden damage to auditory cells, neurons, then they start in their repair phase behave like newborn cells. Basically, they become excitatory, so which means that you get the opposite effect of what you think you have on those cells that are in the repair or recovery phase. And so then, of course, the timing becomes crucially important on when you give GABAergic products, whether it is GABA, and especially GABA-A products like clonazepam, for example. And so in tinnitus, you have the group of Tsanopoulos has looked at this GABA kind of switch-like behavior in the dorsal-cochlear nucleus, where depending on the amount of those channels, you get hyper or not hyperactivity. And so the essential thing is that how well this switch works might actually determine on whether you develop tinnitus or no tinnitus if you have hearing loss. This might not be for every patient the case, because it might only be for those who have a sudden hearing loss or for those who have noise trauma-related hearing loss. Might not be an age-related hearing loss, for example, but it would be nice to have a subgroup where we understand the mechanism of action and can tell, okay, well, if this was the cause, then you should be good by activating GABA if we're there at an early level, or blocking it at the early level or at the late level. So there's the understanding of this switching of the GABA from excitatory to inhibitory might actually determine, first of all, whether somebody gets tinnitus or not, and the second, when we should apply treatments. So I think this might be more important than the general mechanistic view of how potassium is involved in repolarizing depolarized cells, like in any tonic and in any burst firing.
Just a little bit of a commentary here. Acoustic shock has actually also, without hearing loss, has also been shown to, I guess, to kind of launch this process, I suppose, which I guess would in turn explain people who have no hearing loss, also getting DCN hyperactivity, I guess.
Yes. So I assume that any trauma that results in repair will go through these phases. The problem is, how will we visualize it? Or will we just compute it and say, when did the trauma happen? Okay, it happened so long ago, so then we have to do this. If it happened longer, but then we have to really have the temporal resolution of an understanding of when this switch actually occurs. And this is what Arnaud Norena is looking at currently in France.
Okay, excellent.
And then the last question, I think, benzodiazepine use, and benzodiazepines, of course, are related to this GABA A effector. So the question is why. So it has been estimated that 15 to 25% of long-term benzodiazepine users experiences tinnitus as a withdrawal symptom, with those tapering off long-acting agents like diazepam or clonazepam as well, which is very long-acting, being more likely to report tinnitus compared to those on short-acting agents. Given that benzodiazepines are often prescribed to individuals with moderate or severe tinnitus, these numbers suggest a significant risk of tinnitus worsening during withdrawal, especially for those with pre-existing tinnitus. And then, of course, the question is, why has this risk not been more fully addressed in the tinnitus treatment guidelines? And what are your views on incorporating benzodiazepine withdrawal protocols into tinnitus management? So, to be very clear, And the second question then is, how can we better support patients who experience tinnitus exacerbation from benzodiazepine tapering? So, first of all, most of the patients I see get about benzodiazepine, and the one I use most is clonazepam, which has a very long half-life. So it should theoretically be a high risk. Many patients are afraid of the so-called addiction properties of these benzodiazepines. Now, intriguingly, if you use it so first of all, clonazepam is a antiepileptic, and the dose that is given for a treatment of epilepsy can go up to 3 times 2 milligrams a day. The dose that we prescribe for tinnitus is 0.5 milligrams a day, and we try to give it for 3 months, and then we taper it down. The reason why we take 3 months is because that's one of the time frames of creating changes in connectivity in the brain. Now, if you give clonazepam for less than 3 weeks, you can stop from day 1 to day 2, but even then it's better to taper it down. Now, if you we combine this benzodiazepine with Deanxit, which is not available in many countries, but it blocks your dopamine D1 receptors to defocus the brain on the tinnitus. And when people are highly stressed because of the tinnitus and they're in overdrive, then the Deanxit and the benzodiazepines actually relax the hyper-sympathetic activity. But there is a second reason why we give benzodiazepines, and that's because if you give Deanxit, there is a risk of tardive dyskinesias, which is based on the dopamine blocking effect. Now, tardive dyskinesias are treated with clonazepam. So if you already give it from the beginning, I've never in my whole life seen tardive dyskinesias caused by Deanxit when you combine it with the clonazepam. However, I've seen 2 cases of tardive dyskinesias who were not treated with benzodiazepine. So the benzodiazepine has one reason is to prevent tardive dyskinesias. The second reason is it improves sleep, but not deep sleep. It doesn't improve deep sleep. It can improve REM sleep, which is correlated to the loudness. So it has a benefit on that as well. And then the question is, how do you taper it? Well, if you only start at 0.5 milligrams, which is 1/4 of a tablet of 2 milligrams, then you prescribe the 0.5 milligrams, and then you go per week, you remove a quart. And then normally there is no problem. However, some people relapse in the tinnitus severity without the benzodiazepine or without the Deanxit, and they stay on maybe, let's say, 1/2 a tablet of Deanxit every 3 days, and maybe 2 drops of Rivotril. But if you consider that, it's a lot better to stay at these extremely low, almost homeopathic doses of Deanxit and clonazepam than to have high stress levels, which are way worse for your brain physiology. The high stress levels are way worse than those very low doses. So what you should not do is taper it very quickly and give high doses. I think anything which is higher than 0.5 milligrams is probably not needed. And if there is a sleep issue, there is better drugs than benzodiazepines to address the sleep problems. If people have deep sleep problems, you can help them with not with a lot. There's not a lot of drugs that really benefit, but some antihistaminics can do it. Trazodone can give some benefit. For REM sleep, you can use either clonazepam, but on top of that, you could also consider melatonin, which can give some benefit, and cannabidiol, that can also improve it. So there is not that many drugs that do give selective improvement of REM or deep sleep. And how can we better support patients who experience tinnitus exacerbation from tapering? Well, the first is to restart the benzodiazepine and taper it very slowly. Of course, if you see a patient who has been on 3 milligrams of benzodiazepine, then your starting point is already very, very high. And then it should be very, very slow. And on the moment that you see that they're getting trouble, even if they're still at 1 milligram, then you just say, okay, stay on it for another 2 months and then we'll start tapering again. But with low doses, normally it's not too much of a problem. So it's basically making sure, and the good thing about tinnitus is that if you use it for tinnitus, then you never have to increase the dose. If you have to increase the dose, it's better to start trying other medications, because if you go from 0.5 to 1 milligram, that people will need higher and higher doses, and then you're down the rabbit hole. So stick to the lowest dose of 0.5 milligrams if you give benzo. It's like clonazepam with, and then you should be fine. And intriguingly, in the combination with the onset, you don't have to increase the dose. If it works, normally it keeps on working. And usually after 3 months, you can taper it down to half a tablet, first 3/4 of a tablet for a week, then half a tablet for a week. And if need be, then people can stay on these extremely low doses. So don't force to go to zero. If it forces, if they can't sleep anymore, if they get louder tinnitus, etc., you stay at the lowest dose, and then 3 months later you can try it again to taper it a little bit down. So tapering is a matter of having patience and not forcing it in a rapid schedule.
Okay. So as I understand this, this combination of clonazepam with Dianxid is mostly for emotional stability, as I understand. It's not for any subjective or objective decrease in tinnitus. Well, objective, sorry, not subjective.
So when you start with this medication, usually within 3 to 4 days, people feel a difference. And the difference is, so we routinely ask 3 questions. One is, how loud is your tinnitus from 0 to 10? The second is, how much does it bother you from 0 to 10? And the third is, what is the percentage of the time during the day that your tinnitus is dominantly present? The third question is the most important. If somebody has only 10% of the time tinnitus that is present, you can give those 2 medications. It won't do a thing. If, however, it's above 60%, then it means that the brain is focusing a lot on the sound because it's dominantly present. And D1 receptors, there is only one drug that I'm aware of that blocks D1 receptors. That's Dianxid. And so by blocking those D1 receptors, the focus is removed from the tinnitus, but also the focus of pain or the focus on whatever they're hyper-focusing on. This comes at a risk, meaning that if you take it for a long time, also your focus, for example, on sex will decrease because it just blocks every focus. And therefore, after a couple of weeks, you should already think about tapering the dose, which is difficult because it's a very small tablet. So you need to cut it with a pill cutter, or you have to take it once every 2 days. And then usually those side effects also improve. But how do you know when to taper? That's very easy. when somebody has severely bothersome tinnitus with hyperfocus, meaning their brain is in hyperfocus on this tinnitus, then if it works, within 2 to 3 days, it will give defocusing of the tinnitus, so they can think about something else than their tinnitus, and the fatigue or the concentration problems that come with the hyperfocus on the tinnitus will improve because they're defocused from and they will be less tired because of that. Now, the deanxit also blocks your alpha-1 receptors, which are part of your noradrenergic fight and flight response. So it will also calm you down, so you'll be fighting the tinnitus less. Now, that being said, if you take it and your system has normalized, meaning you're out of this constant fight and flight mode, then of course, if you take the same dose, you will become tired. So, and then when the patient says it's strange, around 1 o'clock, in between 12 and 2, I have to take a nap. And I never used to take a nap. Then it's the alpha and alpha-1 and dopamine D1 blocking that is causing it. So then I have to start tapering it because I get tired. So it's very safe and very easy drugs to monitor. As long as you know what you're doing, it's a very easy and safe drug, and it acts very quickly. It doesn't have a lot of side effects at these low doses, except if you have to give it for a very longer time, then you do get some libido decreases, and if you have to give it for a longer time, then people also say, well, I'm not very emotionally involved anymore, because of course you block every focus and every arousal, which is essential for emotions. So you don't give it for years for most people, except if they have a genetic polymorphism of their dopamine receptors. So that being said, the last question is, what's the most promising?
Sorry, really quick before we get to the last question, I have a question about the clonazepam. So basically, would you recommend this combination of drugs for people who are already in a very, very excited state, for example, or a very heightened level of hyperactivity, and I assume that people who are in a very high level of hyperactivity have significantly higher chances of having negative reactions due to the tapering process. So if this combination is predominantly used to try and maybe desensitize or to remove the attention from the tinnitus, would it be better for people with a high level of severity to steer clear of it, or to try and control their emotional reaction to tinnitus in different ways?
No, because of your alpha-1 blocking, but then I would add catapresson or clonidine, which blocks your alpha-2 receptors clinically. It's actually an alpha-2 agonist, but it's functionally, because of the location of the receptors, because they're presynaptic, they're actually blocking the alpha-2 activity. And so then you block your fight and flight response, which means you remove the hyperactivity by the combination. And then it is actually a good treatment for those people. But again, it's not because it's very heightened activity that you have to increase the dose. No, you stay at this very low dose. It can take maybe a little bit longer, but there's no need to increase the dose because of the severity of the symptoms. Either it works in this low dose, but if they're in an extremely heightened stress-like situation, then and that would impede their sleep, then you could add a little bit of an alpha 2 blocking, well, functionally alpha 2 blocking mechanism like clonidine.
Okay. All right. Well, I guess let's move on.
So then what's the most promising approach? Well, as we discussed earlier on, probably we need to find a brain signature of tinnitus, which is what of tinnitus itself, the presence or not, and as well as the intensity or the loudness and the amount of distress. And then the idea is a multimodal approach. Well, we know based on meta-analyses which parts of the brain are involved, and we can theorize then a model here where we have an increase or the lateral pathway which processes the loudness, and the medial pathway which processes the suffering, and then the descending inhibitory pathway. So we know these areas. And then the question is, okay, well, we should activate the descending pathway, we should activate inhibition, and we should block the medial pathway, and we should block the lateral pathway on the condition that there is one final common pathway, which is the balance between these two and the descending pathway. Now, from a practical point of view, if you put all the meta-analyses together, well, we know all these areas that are involved. And the question is, well, we can't just modulate all of them at the same time. Therefore, we have to look at how these areas are connected based on the complex adaptive system idea, that the emergent property, like in the transformers, where the different building blocks are connected in a different way, and that one connectivity profile will be the tinnitus network. So we have to consider modulating a network. And how do you do that? Well, just like in war, you find a battle with combined arms, meaning the Air Force, the ground troops, and the Navy, which is called a joint airfare. We can also do multimodal treatment, where we use what we know from auditory treatments, pharmacological treatments, neuromodulation treatments, physiotherapy treatments, psychology treatments, and we can start combining these within a domain. So we can do hearing aids and sound therapy, we can do medication, psychedelics, we can do lots of different neuromodulation approaches, physiotherapy, different kinds of psychology, or we could go multimodal, meaning that we combine auditory with pharmacological, with neuromodulation, with physiotherapy, with psychology in different forms and variations. And this is similar to having an army consist of the ground troops, the Navy, and the Air Force. But the problem, however, is how to study these scientifically, because we are used to study hearing aids individually, or one medication individually, or one neuromodulation individually. So we know that if we remove it or do a placebo for it, it's beneficial. Whereas if we start combining all these things, it becomes scientifically more difficult. And this is where I think we should probably move to what we mentioned earlier on, and that is a mechanism-based approach, where we really look at which mechanisms are complementary. So, for example, from a Bayesian brain point of view, hearing aids and sound therapy combined make no sense, because the brain predicts sound will arrive. If you have hearing loss and the sound doesn't arrive, and your brain is in a stress modus, it will say, better safe than sorry. I need to get this auditory information that's not arriving, so I will generate it myself, and it fills in the missing sound constantly, and that's where you get tinnitus. Now, the way that the prediction works is that it tries to pick it up by strengthening its pickup potential, and by changing the gain, which could be hyperacusis, so which initially might be beneficial. But of course, if you're simultaneously going to present sound therapy, you actually prevent the brain from still picking up the missing information. And therefore, if you look at studies that combine hearing aids with sound therapy, there is no added benefit from the combination to the single, because theoretically it doesn't fit. Whereas combining, for example, psychedelics, where we know they massively increase plasticity, and then using neuromodulation to drive the plasticity in a certain way, that theoretically makes sense. And so that's where, if we start with this multimodal approach, we have to have good theoretical models and mechanistic approaches that say, okay, this and this combination is good, but that and that combination is probably not good. That doesn't mean you don't have to test it, but it does help from a mechanistic point of view to know which combinations are good. For example, if you take excitatory medication like methylphenidate or modafinil, then you increase your excitability, which might be beneficial for using TMS afterwards. Or if you use D-cycloserine, which increases your glutamatergic long-term potentiation, then if you want to change a pathological pathway, then combining that with rTMS has shown that you can double your effect size. So there is certain combinations that are better than others. But I do think that if we consider tinnitus as an enemy, then we have to fight it with an army of whatever arms we have, and that's going to be multimodal, but in a rational way, not just throw everything at it at the same time, because that won't work. But in a mechanism-based approach, say, okay, well, these and these combinations do seem to work. And for most of the patients that we see with severe tinnitus, they do end up with a multimodal therapy that can consist of hearing aids with medications and neuromodulation, with some trigger point treatments. And if they haven't had, and if it's trauma associated, we might propose EMDR. So we do use the full spectrum, and not for everybody. It's personalized, but it does make sense from a philosophical point of view to try it in patients with severe tinnitus.
Okay, what an excellent description. Yes, I absolutely agree that a multimodal approach would show more efficacy rather than focusing on one single one. I guess that's pretty much it for the presentation, as I assume. Okay, well, let's move on into some extra questions. This should not take too long, but just some minor ones that have arisen. Now, one thing I would like to discuss just very briefly is some of Jasper Bob's opinions that he shared in his book, Tinnitus for Training Therapy. And one thing that I noticed he mentions pretty confidently, and by the way, this opinion has spread throughout the world of audiology and ENTs. So pretty much absolutely everybody in a clinical setting believes this. But so what this belief is that being in silence is unnatural for the human body. He gives like a interesting description that the body must always have some sound for it to be natural or something like that. And that being in silence apparently worsens tinnitus objectively. Now, the problem I have with this opinion is that it goes against all logic and I guess all scientific logic of spike timing dependent plasticity, long-term depression, long-term potentiation. A person being in silence should have decreased stimuli, which then could lead to reduced calcium influx, which then the brain interprets as the synaptic connections being unimportant and potentially actually leading to their to the AMPA receptor removal, for example. So I wanted to hear your opinion on this. Being in silence worsens tinnitus, and whether or not it has any, any place in the community, the scientific community?
Well, if you look at it, I'm a Bayesian person. So if you're Bayesian, if you believe that the Bayesian brain is an explanation, then when you have hearing loss as a trigger for, which is not for everybody, but let's say it's one of the most common associations. If you have hearing loss, let's say you've been to a concert, you've got a drop in 4,000 or 6,000 hertz, you've got a dip, and that dip corresponds to a 4,000 hertz tinnitus match sound of 5 or 10 decibels above hearing threshold. Then, of course, the filling in mechanism is because your brain predicts the 4,000 hertz should be there. It's not there, so it will fill it in. Now, if you're in silence, then of course there is certainly not going to be a 4,000 hertz sound to be heard, and therefore the brain will certainly fill it in. So from a Bayesian brain perspective, being in silence is not optimal. If you're, however, in a little bit of a noisy environment, what will happen is that because of the tuning curves, the tuning forks will actually widen when in order to try and pick up this missing information. So if it's a very sharp hearing loss, then the tuning forks widening might actually cover the area, and that might be better. So from a Bayesian point of view, it does make sense that silence is suboptimal for certainly in the beginning phase for turning acute tinnitus and chronic tinnitus. Of course, there might be different explanations if you have a more psychological approach to it. Then you might say, well, if you're in silence, you will focus more on the tinnitus, and this might be worse. So it really depends on the model that you have of how tinnitus is generated. And remember what I told earlier on about the 3 paleobiologists who, based on the same data, came up with 3 very different explanations. And so until we know which model is the best model, then there might be different explanations that actually come up with very different reasoning. And ultimately, from an evolutionary point of view, the model that will survive is the model that can explain most. And if you have one model that can explain everything about tinnitus, both tinnitus associated with hyperacusis, with Noxacusis, with depending on different contexts, if you have one model that can do it, well, that's a model that we should follow. Up to now, the models have changed constantly. Initially, people thought, well, tinnitus is in the ear, so the only treatment was hearing aids. Then Rodolfo Llinás in the 1990s said, no, it's in the auditory cortex. So we targeted the auditory cortex with the MS and TDCS, as I mentioned earlier. Then when he says it's a network phenomenon, so we start treating networks. When we suggested it could be a Bayesian problem, then we said, well, how does that lead to a treatment? And we had no clue how it could lead to a treatment because we didn't understand the Bayesian mechanisms in the brain. Now we know that the predictions are predominantly in theta and in beta, and prediction errors in gamma, which is in keeping with the thalamocortical dysrhythmia. So we see that Bayesian and Bayesian modeling and thalamocortical dysrhythmia seem to align. And so depending on which model you use, and the models are constantly evolving, ultimately it will depend on the best model, not the best, the most fitting model with the clinical data that will turn out to be the one that we should all follow. But until that moment, depending on your background, of course, if you're a psychologist, you will have a different model of thinking. And some psychologists believe in Yosubov's model, which is purely based on conditioning. And others says, no, I don't think it's conditioning. And because of this and this reason. And so, you can't, until the moment that we have one generally accepted model of how tinnitus arises, there will be different explanations, and the most fittest will survive.
Yeah, the biggest issue I have with Jastrabov's model is that, from my understanding, is basing it pretty much exclusively on assumptions. And of course, the data that I have collected over the years of my research, of course, it's purely anecdotal, has actually shown that severe to catastrophic sufferers can actually see a reduction in their symptoms after months and months and months in silence. Unfortunately, yes, until we have some more clinical data on this. Now, you normally do not have people who are maybe suffering mildly or moderately staying for months in silence. That just doesn't happen. People don't do that. And I'm certainly hoping that in my career that I will be able to organize a study such as this, but nobody of moderate to mild severity wants to stay in silence for many months to try and prove it. And maybe fortunately,
No, but even if you could do it for a subgroup of severely suffering patients, then, well, you can't generalize your model, but you can still say, well, for this subgroup, this seems to be beneficial. And then we just have to find an explanation why it might be beneficial for one subgroup and not for the others, or we just have to admit, look, we don't have the data yet, so we can't say. But then that's where theory comes in, and you have to say, well, if this is the model, then if we would generalize it, would it also be logical to apply it to people who do not suffer that much? And then you have to find the theoretical model that can also explain, but the mechanism of action doesn't have to be the same for everybody.
absolutely, I agree. I guess it really comes down to like balance, whether or not a person might see or deem it worthwhile staying in silence for months on end just to get some clinical data. I think that people who suffer mildly and moderately might see that as a larger issue rather than, kind of plowing through it and just enduring it as it is. Okay. Well, I guess there is actually one last question that some people have wanted to ask, and that is about norepinephrine. It has actually shown also anecdotally to help hyperacusis and even Noxacusis, as I understand. And I wanted to see if there might be any explanations, or you might have any ideas as to why that is the case.
Yes, of course, this is very counterintuitive because normally your alpha-2 receptors are predominantly which are the ones that norepinephrine works on, are predominantly located in your sensory cortex, so your auditory cortex, somatosensory cortex, visual cortex, etc. Now, norepinephrine has what's called an inverted U curve. So at a very low dose, it doesn't do anything. At the optimal dose, it will increase sensitivity, and at a very high dose, it will block the opposite. And that's why if you are when an animal is in stress, it will either fight or flight. That's the typical fight-flight response. Or if it's in life-threatening stress, if it doesn't see a way out, it will pretend to be dead. And that's because of the inverted U curve. So at high dose and at optimal dose, the effects are actually the opposite. And it's the same for dopamine. It's for everything that is stress related. And that is just to make sure that if there is no way that fighting or flighting would be better, then you just freeze. So that is probably the reason why you can hear very different results of giving dopamine agonists, dopamine antagonists. And you say, well, that's strange. Both an agonist and an antagonist works, or noradrenaline works, and then noradrenaline blocking also works. And that's because of the dose effect. So normally, when you're in stress and you predict something is going to come, then you want to pick up as much information as possible. So you're walking in the dark in a forest, then you want to pick up, because you don't see very well, you want to pick up every sound that is possibly available. So you're going to change your sensory gain by your alpha-2 receptors. So your noradrenaline, which will be pumping high if you're stressed because you're fearful in the dark forest, will actually increase your sensory gain. The problem with increasing sensory gain is that you're going to pick up things that might not even be there, because your sensitivity is so high that you won't pick it up. And that's why normally in stress, your tinnitus gets worse or your hyperacusis gets worse. But if you're in life-threatening stress, or your noradrenaline is going to be so high that it will block everything and you'll freeze to death and be so scared that you won't move anymore. Now, without stress, if you give noradrenaline, it can do the same thing. And so and noradrenaline also improves sprouting. It improves dendritic sprouting. So it improves the sensory nerve fibers to go and pick up more information. So that might be a second mechanism, but this is way slower, of course. This takes at least through 3 weeks to 3 months before you can see that effect. So in the acute stage, it depends on the dose because of the inverted U curve. And the chronic stage, it might be beneficial because it increased sprouting, which is normal, because if you're stressed, your brain wants to rewire so it can pick up even more information from a missing area.
Well, okay, I guess that's pretty much it.
Are you sure you don't have more questions, Anthony?
Honestly, at this moment, I'm sure I do, but it's been quite some time.
The ride, yeah.
Yeah, I don't have neurosurgeon-esque stamina, so I guess we can finish up for now. I guess, and I'm sure if that more questions arise in the future, I'm sure that future Q&As definitely can address them. But I think that the questions that were asked today and the answers that were given and the wonderful presentation that was made by Dirk, absolutely phenomenal. I can say absolutely huge props to you all for allowing this to happen, and I think that this Q&A was very massive for the community, I think.
Yeah, thank you, Anthony, also for preparing this so diligently.
It was an absolute pleasure putting all these questions together, not only from myself, but from the community as well. And I definitely hope that most of the people who participated in this process got the answers that they wanted to.
I hope so, yeah. Well, Dirk, I want to give you a huge thank you. So the viewers and listeners might not realize this because, of course, we're going to be editing this session, but you've spent now almost 5.5 hours with us this evening. It is after midnight, you're in your time zone. And we actually had a failed recording attempt last week. Plus, you must have spent a lot of time preparing these slides. So I just want to thank you for the huge investment that you've made that really shows your commitment to, communicating with the tinnitus community and, connecting with people. We really appreciate it.
Well, you're very welcome. And I do think it's our part of our task. We get, as academics, we get funded by society. I mean, all of you pay for my salary in a certain way. So I think it's absolutely normal that we give back to society what we get from society to allow us to do research, and it's a normal part, I think, of our life that we because we are paid by all the citizens that we give back to people what we want. And I also think it's very important for people to understand that unfortunately most of the research that we do might actually be not successful, and unfortunately this does not get published because it's very hard to publish unsuccessful data. And I can understand that a lot of people feel frustrated because the perception is, well, there is no news, so nobody is doing anything. Unfortunately, we're doing. There's many research teams like ours who do work, but not all negative results are. Well, most of the negative results are not published, but that doesn't mean we don't learn from it. As I mentioned, a model improves by seeing why it does not work, and then you have to adjust the model. So progressively, there is going to be an improvement. And of course, the more people do research, the more negative results we have, the quicker we will get to solutions. And this is the way science work. We do not learn a lot from positive results. We learn more important information from what does not work, but that unfortunately doesn't get published.
Yep, I love that attitude. Well, again, a huge thank you to both Anthony and Dirk. A thank you to everyone who listened to this, and I hope it's been really helpful to you. If you did like this, we'll definitely consider doing more of these in-depth Q&As in future. If you want to discuss this more, you're welcome to do so on Tinnitus Talk. If you want to support innovative, cure-focused tinnitus research, of course, please do visit tinnitusquest.com and consider making a donation. Thank you all so much, and we hope to see you next time.